Good morning, everybody, and welcome to the third day of the Mycokey final conference. I would like soon give the words to Antonio Greco that will introduce the last day of the conference.
Thank you very much, Antonio, and welcome to all the audience and all participants. So this is the last day of Mycokey conference online, and I'd like to introduce just an important section that we are going to listen is food and feed remediation and purchasing. And the second one is modeling and ICT solutions that are from outcomes of the project. The project important results that we got from the project. Then we move also voice and feed industries again, and then so we had good talk, particularly with some partners, industrial partners that we had a really great collaboration during the project. Finally, we have a sign from a European project and the strategies. We'll get the chance to listen Rudolf Kersker, who is coordinator of My Toolbox, then Alessio Bacchielli from the European Commission, and finally Franz Versate, also from the European Commission. So thank you again for your listening for this third day, and then so go ahead, Antonio, for your presentation, for your introduction.
Thank you very much. Now we have the first speaker, Michelangelo Pascale from Italy. He is the leader of Work Package 6, mycotoxin remediation. So he will introduce the main outcomes from the Mycokey project for this work package. Michelangelo, please, the words are yours.
Thank you very much, Antonio, for this introduction, and hello to everyone. Can you see my screen?
Yes, yes, I can. Well,
okay.
Actually, I suppose everybody.
Okay. I will talk about the main results obtained in the work package number 6 of the Mycokey project, and they are relevant to the strategy for remediation of feed and food commodities. As we know, mycotoxic contamination of food and feeds is a worldwide problem with serious public health and trade implication, and with serious economic impact. Any strategy for preventing and reducing fungal and mycotoxin contamination must be carried out at an integrative level along the food and feed production chain. Practical interventions are carried out prior to fungal infection or during fungal infection of plant material by means of good agriculture practices and good storage practices. However, under certain environmental conditions, the contamination of crops with mycotoxins and fungi could be unavoidable. Remediation strategies are necessary in order to recuperate some lots and batch. What are the possible solutions when the level of contamination of material, food and feed are higher than legal limits? We have several solution. The first one is blending of non-contaminated material with material above the limits, but this solution is prohibited in the European Union. Another solution is the destruction of contaminated products, but there are several economic losses. Another one is the use of these contaminated lots for biofuel production, or depending from the level of contamination of these commodities, the redirection into feed for less susceptible animals, and the use of feed additives is advised. And the last one, this is the topic of the World Package 6, is to develop strategies of decontamination, detoxification, in order to recuperate commodities contaminated by mycotoxin and avoid economic losses. The main objective of World Package 6 are to develop practical and effective post-harvest procedures for mycotoxin reduction in the food and feed chains, and also to provide alternative and safe use option for mycotoxin contaminated batches. The work package was organized in 4 tasks. Each task has a specific objective. Task 6.1, the objective was the improvement of post-harvest cleaning technologies for mycotoxin reduction in maize. The second was the development of new feed additives, but additives to be used as multi-mycotoxin detoxifying agents in order to prevent, reduce mycotoxin exposure in farm animals. The third task was the selection of microorganisms and enzymes for biodegradation, biotransformation of mycotoxins contaminated feed and food. And the last one was the safe use option for mycotoxins contaminated batch by optimizing fermentation processes for biogas and bioethanol. 4 key actions have been explored within this work package. The first was the clinic technologies, but at industrial scale level. The second one was the feed detoxification by developing new feed additives. The third was microbial and enzymatic detoxification, biological detoxification. And the last one was sustainable fermentation processes by the production of biofuel. The partners involved in this work package were 4 from Europe and 3 from China, belonging to universities of public institutions, and 4 industrial partners. One of these left the consortium in November 2017. The first one. We know that maize is the most important food crop in the world. However, it is susceptible to mycotoxin contamination. And also, it's well known that moldy kernels, discolored kernels, and injured kernels, or small broken maize kernels, are very contaminated by mycotoxin. A combination of clinic technologies to remove this contaminated fraction can reduce the mycotoxin contamination in the final product. in the Mycokey project, two case studies have been performed in Germany and Spain in order to verify, to assess the efficacy of industrial cleaning equipment for the reduction of aflatoxins in maize and for the reduction of the major fusarium toxins in maize. And the sampling was performed according to the Commission regulation relevant to the sampling procedure for big lots of cereals. The first case study, a reduction of from 55 to 94% of aflatoxins level in maize was observed by combining the mechanical cleaning and expiration, density separation, and optical sorting. And the rejected fraction was about 10%, and the rejected fraction were contaminated with aflatoxins at very high levels. And the results show that by using this industrial cleaning technology equipment, it's possible to convert biomass and feed quality maize in the feed or food quality maize with significant economic gains for the farmers. The second case study was relevant to the fusarium mycotoxins reduction in maize, deoxynivalenol, zearalenol, and fumonisin B1 and B2, and again an overall reduction of DON, zearalenol, and fumonisins ranging from the value that you can see in the slide were observed, and again, in the rejected fraction were observed very high level of mycotoxins. The rejected fraction was about 4–6%. That means that the incoming product was very clean. And you can see that the level of mycotoxins in the incoming product were quite low, below the legal limits. However, the technology are very good because in a previous study we observed with very contaminated maize, raw maize, by deoxynivalenol, zearalenol, and fumonisins, the same or higher percentage of reduction. However, you can see that the percentage of the rejected fraction was very high, 17%. The second key action was the detoxification. And mycotoxin, the research activity within this task were focused on the in vitro screening of materials in order to verify the efficacy in reducing simultaneously target mycotoxins, in particular aflatoxin B1, zearalenol, ochratoxin A, and fumonisin B1, and to evaluate in vivo the efficacy of the best selected feed additives in reducing mycotoxin absorption in target animal species. In addition, has been optimized and validated in Ispa laboratory a new protocol for the selection of feed additives to be used as mycotoxin detoxified agent. In the European Union, bentonides are allowed for mitigation of aflatoxins contamination for ruminants, swine, and poultry. And a key point of this regulation is that bentonides should demonstrate an aflatoxin binding capacity higher than 90%. The binding capacity is determined by a method that has been developed and validated by a single laboratory, and that has been approved by the European Reference Laboratory for Feed Additives. The standard operating procedure are reported in the slide. We did a Mycokey project, has been evaluated the robustness of this official adsorption assay, and Ispa proposed a new and optimized protocol by varying some parameters in terms of sample preparation, incubation time, buffer concentration, organic solvent percentage, and centrifugation condition. The protocol has been validated in-house, and the experimental results obtained by the validation study showed satisfactory precision with a maximum relative standard deviation for intermediate precision of about 3%. This optimized assay was applied to several smectites, and you can see the comparison between the official method and the optimized assay. You can see that by using the European Union method fixed by the regulation, several bentonides can be erroneously excluded from the market. This method will be submitted to the European Reference Laboratory for Feed Additives for its approval. This is the next step. We did a Mycokey project. Several materials have been tested in order to verify their ability in reducing simultaneously the major mycotoxin, aflatoxin B1, zearalenone, fumonisin, T2 toxin, ochratoxin, and I will present just some of the main results obtained in this task. And the first one is development of a new feed additive containing sodium smectite and a biopolymer very rich in polyphenols. You can see from the figure that a very high mycotoxin absorption percentage was observed at several pH for aflatoxin B1, fumonisin B1, zearalenone, T2 toxin, and ochratoxin A. And these the other good results is the development of a new feed additive containing an acid-treated durian peel. This result has been obtained thanks to the Mycokey Fellowship for Short Learning Visit that a PhD student from the Thammasat University in Thailand was in our lab, and the collaboration was very fruitful. And this new feed additive has been tested, and a very high mycotoxin absorption was observed for simultaneously for aflatoxin B1, zearalenone, aflatoxin A, and fumolysis B1. Within this task was developed and scaling up at pivot level a process to produce a new bioorganonuclei acting as multi-mycotoxin adsorbent. This is a sodium bentonite that has been activated, and the surface was modified. And in vitro, it was observed a very high absorption, this modified bioorganonuclei compared to the native bentonite, native native clay for aflatoxin B1, zearalenone, aflatoxin A, and FB1. And this bioorganonuclei was assessed in vivo experiment in order to verify the efficacy in reduce the exposure to aflatoxin B1, zearalenone, aflatoxin A, and B1 in rats and piglets. And the next talk, Vito Dascano will give you more details about these results. Another good result was the development and scaling up at semi-industrial level of a new process to produce a yeast cell wall-based product for multi-mycotoxin absorption. And it was verified in vitro experiment and showed good, for the first time, the simultaneous absorption of zearalenone, aflatoxin A, and fumolysis B1, and less was the absorption for aflatoxin B1. And again, this material was tested in vivo experiment, and the Virginie Marquise will talk about this novel feed additive in his presentation after the coffee break. Another task was the microbial and enzymatic detoxification, and several activities were performed in order to develop new biological agent for detoxification of feed. And the first activity was to optimize the production of protease for degrading of aflatoxin A during wine processing. And results are summarized in this slide. 3 aspartyl proteases genes and 2 serine proteases genes of Aspergillus carbonarius involved in OTA degrading activity have been identified and characterized. The recombinant vector for the production of Carbonarius proteases was cloned in Escherichia coli and Saccharomyces cerevisiae, and these cloned microorganisms showed increasing proteolytic activity. And one strain of Lactobacillus rasmosus showed higher than 97% of total reduction, and one strain of this fungus, this yeast, and Mycokey pulcherrima, and 3 strains of Lactobacillus plantarus were used in a lab scale wine making, and a significant reduction of aflatoxin A was observed in red wine and must. Another good result was obtained by Professor Liao of Huaqiang Agricultural University in China. He will talk about these results in his presentation later. And just I would like to highlight that a new bacterial strain of the genus Phingomonas was able to degrade DON of 100%, and the same bacterial strain was able to reduce triacetyl-DON also. And the degradation, that was biotransformation products, were characterized by several analytical techniques, and they were 3-oxodeoxyrivalenol, 3-epideoxyrivalenol, and 3-epi-DOM1. Another good results was obtained by the Yas University, Yas in China, the Jiangsu Academy of Agricultural Sciences in China, and again a new bacteria was identified as the Rosia insula, was is isolated and characterized, and was able to degrade DON up to 88% in a liquid medium. Again, the biotransformation product was identified as 3-keto-DON, that is less a metabolite, less toxic than the oxynivalenol. And this bacteria was able also to degrade triacetyl and 15-acetyl-DON. Another activity was relevant to the detoxification of mycotoxins contaminated cereals by Pleurotus species, and it was observed a good reduction of aflatoxin B1 by this fungus in liquid and solid media, but also in industrial mushroom growth medium. And a good result, reduction was of monocytes B1 was observed. And in order to explain the mechanism of degradation, it was observed that part was due to mycotoxin sorbent. The reduction was due to mycotoxin sorbent, and the mycelium of this Pleurotus was able to remove up to 85% of aflatoxin B1 in buffer at different pH. Also, an enzymatic degradation was of aflatoxin B1 was observed by using crude extract from spent mushroom substrate of Pleurotus eryngis, and was purified the enzyme lactases from this Pleurotus pulmonarius and Pleurotus eryngis, and this lactases, this enzyme, was able to degrade zearalenone, aflatoxin A1 100%, aflatoxin B1 90%, fumarate B1 70%, T2 toxin 40%, and ochratoxin A 27%. The degradation biotransformation product of aflatoxin B1 were investigated by high resolution mass spectrometry, and this enzyme was able to hydroxylate aflatoxin B1 to aflatoxin 2,1, that is a less toxic metabolite. This activity was performed in collaboration with Agriculture and Agri-Food Canada, Dr. Mark Soumaran. And the last key action was relevant to the fermentation, the biofood production. And the activity were relevant to production of biogas and production of bioethanol. And the main results obtained during the activity of biogas production were the development of a new method for the evaluation of mycotoxin contamination in the digestive, identification and taxonomic classification of the microorganisms involved in the biogas production, and were also identified the functional activity of the main microbial population during biogas production by means of metagenomic analysis. And the biogas production has been observed that was not affected by the level of the presence of mycotoxins in contaminated biomasses, and has been observed after 15 days post-inoculation in a lab-scale bioreactor, the 54% of the reduction of aflatoxin and 60% of reduction of human CB1, and the metagenomic analysis revealed the presence of 2 new carboxy X-rays involved in fumonisins degradation. Concerning the bioethanol activity for the production of bioethanol, were screened different yeasts in order to evaluate the tolerance to complex medium and different mycotoxins, and has been observed that some yeasts were tolerant to fumonisins B1 up to 60 ppm, CRR up to 50 ppm, were lower tolerant to T2 toxins, but of these produced ethanol from maize materials contaminated with aflatoxins. And another good result is that a new engineered yeast strain producing lactases has been obtained for mycotoxins degradation or biotransformation. And the Saccharomyces revisions from the VTT collection, culture collection, was selected based on its tolerance to acidic condition, a different amount of mycotoxins, and also based on the presence of inhibitors in lignocellulosic biomasses and as the productivity of bioethanol. And in a lab-scale fermentation process, by using this yeast, a good yield and productivity was observed from a mycotoxin contaminated lignocellulosic biomasses. And in conclusion, I would like to highlight which kind of innovation, which novelty we obtained with these work packets. The first key action, cleaning technology, we have demonstrated, provided that cleaning technologies based on mechanical or optical sorting are effective for reduction of mycotoxins, also in maize, also at industrial level. Concerning the feed detoxification, new technological additives for feed have been developed, and they are multi-mycotoxin detoxifying agent, and 2 anterior search for parting are in progress. And in addition, an in-house validated protocol for the selection of feed additives, in particular bentonite, to be used as aflatoxin B1 detoxifying agent has been developed. Microbial and enzymatic detoxification, new biological detoxifying agent has been isolated and characterized. and an effective biological method for mycotoxin detoxification, degradation of potential material have been shown. And concerning the biological detoxifying agent, a national patent is pending, is in China. The last is the biofuel production. We have selected a microorganism tolerant to mycotoxins, and alternative solutions are possible in order to valorize agriculture wastes contaminated with mycotoxins. This is the last slide that summarizes the results obtained in work package 6 in terms of technology readiness levels, and the key exploitable results. And you can see that the industrial filtering technologies and the new feed additives are reached very high TRL, and you can see more information about these results also on the Horizon Results platform. And finally, I would like to thank, of course, Dr. Logrieco, the coordinator of the Mycokey project, and Nunzia Cito, the project manager. I would like to thank Catherine Beautin of LESAF International, that she has been the work package sub-leader until December 2019. I would like, of course, to thank all task and sub-task leaders with whom I have been in close contact during these 4 years of the project. And I would like to thank all people involved in work package 6 activity. Of course, I thank you for listening. I think that a lot of study is to be performed, to be carried out, because I explained that these results are at lab scale level, and we have some problem with industrial partner for the biogas production, because he left the consortium, and he was the industry that produced at the industrial level the biogas, and this was the reason, because all activity relevant to biogas were performed in lab. But I'm confident that the bioethanol production by the selection of the yeast from the VTT collection is very promising results, and also the engineered yeast could be very promising results in order to use these new yeast to obtain good yield of bioethanol, but also to degrade the amount, the level of mycotoxins in the incoming material. And this could be a good strategy in order to obtain DDGR's the bio product from bioethanol industry with the level of mycotoxins, low level of mycotoxins.
Okay, thank you very much, because a very exhaustive answer, a very nice presentation. I think this kind of study must be really increased in order to also match the new perspective of European Union about the Green Deal that all of us are aware. Now I before giving the words to Vito D'Ascanio, that is the next speaker, I would like to just communicate that the participant to the conference, the registered participants are over 350, with also 60 visualization live on YouTube, and more than 900 visualization on YouTube during these 2 days. So the conference is really getting a lot of interest among the experts of people working on mycotoxin. Now, I introduce Vito D'Ascanio. He will speak about the development of a new bioorganoclay for mycotoxin decontamination, in vitro and in vivo evidence. Please, Vito.
Good morning, everybody. Can you hear me?
Yes, perfect.
Okay, can you see my screen?
Absolutely, yes.
Okay. Good morning, everybody. It's a big pleasure to attend at this international conference, especially as a speaker. In my talk, I will present our work, the development of a new bioorganoclay for mycotoxin decontamination. This work was performed inside the work package 6 of the Mycokey project, and its aim was to develop and to test in vitro and in vivo a new bentonite-based material named bioorganoclay, acting as multi-mycotoxin binder. What about bentonite background? Bentonites are clay minerals belonging to the phyllosilicate class and to the smectite group. They are used in different fields of science, in civil engineering, in medicine, and in a lot of industrial issues. But the use of bentonite as a feed additive was regulated in Europe in 2013, and bentonite was defined as substance for reduction of the contamination by aflatoxins for ruminants, poultry, and pigs. As known from literature, bentonite is effective in absorbing aflatoxin B1 and in preventing the adverse effects in animals and humans. But it is ineffective in binding other structurally different mycotoxins, such as zearalenone, ochratoxin A, or trichotesis. For these reasons, the aim of this work was to chemically modify and to prepare a new product called bioorganoclay, able to bind a large spectrum of mycotoxins at physiological pH values. So the production of our bioorganoclay followed different steps. First of all, the selection of raw bentonite. This step is very important. Indeed, in our recent work published last year, we underline the importance of the geographical and, above all, the geological origin of bentonite in the mycotoxins absorption process. So after the selection of raw bentonite, it was acid activated and then functionalized with an organic modifier. New bioorganoclay was then in vitro and in vivo tested. What we can say about bentonite modifications? The activation process in general consists in a cation exchange between cations presented in the interlayer space of bentonite and cations provided by an external reagent. In the acidification process, the external reagent was an acid, organic or inorganic. In the functionalization process, that was similar to acid activation, the external reagent was a small organic molecule. These two processes were optimized in laboratory, in the first case for the acidification using different type of acid, different acid concentration, different contact time between native bentonite and acid, and different temperature of the process. For the functionalization, we tried different type of organic modifier and different modifier concentration. The experimental condition underlined in the slide were the optimal used to produce the new product. The effectiveness of these two processes was verified by X-ray diffraction and X-ray fluorescence analysis. This new product was then tested in vitro. So we performed mycotoxin adsorption trials using an adsorbent dosage of 0.1% weight per volume and a mycotoxins concentration of 1 microgram per milliliter. As you can see from the graph, the new bioorganoclay was able to increase the absorption values of zearalenone and ochratoxin A at pH 7 and pH 3, and to increase the fumonisin B1 absorption values at pH 7 comparing to the native bentonite. In general, this new product was able to bind more than 80% of all mycotoxins tested, despite two pH values. After that, we did a deep dive inside the variables that could affect the adsorption process. First of all, the dosage effect. So we performed other mycotoxins trials using a different bioorganoclay dosage in the range from 0.0005%, so a very low dosage, up to 0.5%, and performing the trials again at pH 3 and pH 7. As you can see, for all mycotoxins tested, there is an increase in mycotoxins absorption values, increasing the bioorganoclay dosage. So then, using the best dosage obtained in this study, we evaluated also the effect of pH in the adsorption processes. The concentration of mycotoxins in this case is the same of previous study. We can observe that for the mycotoxin adsorption trials obtained in a range of pH from 3 to 9, for all mycotoxins, we observed an adsorption values of almost 100%. Another interesting results were obtained with the desorption studies. With this part, we wanted to understand the strength of binding between new bioorganoclay and mycotoxins. So we performed the adsorption trials at pH 3 and followed by a desorption in neutral pH and in an organic solvent. A change of medium pH from acid to neutral did not release the adsorbed toxins. And in particular, as you can see from the graph, aflatoxin B1 and fumonisin B1 stayed bound on new bioorganoclay also after an extraction with methanol. These results underlined the good binding between bioorganoclay and mycotoxins. To finish the in vitro part, we performed the adsorption isotherm studies. This part is very important. In fact, isotherms were used to calculate important adsorption parameters, such as maximum adsorption capacity and affinity, and to study the thermodynamic and the spontaneity of the mycotoxins adsorption processes. Concerning adsorption parameters, we fitted our experimental data with different mathematical model, Langmuir, Sips, or Freundlich model, and we found that maximum adsorption capacity values determined for aflatoxin B1 and fumonisin B1 were higher than zearalenone and ochratoxin A, and these results were in line with the previous screening. Concerning thermodynamic, I don't have much time to go deeper this topic, but anyway, we performed different isotherms at different temperature, and following Gibbs-Helmholtz equation, we calculated the Gibbs free energy for all mycotoxins adsorption processes. We found a Gibbs free energy lower than 0 in all cases to underline the spontaneity of this process. After that, we evaluated the performance of this new product in vivo in 2 animal models, in rats and piglets. Concerning the rats trial, these trials were done in collaboration with the University of Bari, Department of Pharmacy. We used 12 rats sexually divided, and we provided them a single intragastric bolus of each mycotoxin, with or without the new bioorganoclay. We collected urine samples at different time points, and we analyzed the amount of mycotoxins inside these samples using UHPLC method and following the biomarker approach. The amount of mycotoxins was normalized for the content of creatinine. These toxicokinetic studies provided us important toxicokinetic parameters, such as area under the curve and maximal urine concentration, useful to compare control group and bioorganoclay treated group. So what about results? In all cases, we found AUC reduction for mycotoxins comparing control group and bioorganoclay treated group. Even if we found a statistically significant difference for aflatoxin M1, that was the biomarker of aflatoxin B1 in urine, for ochratoxin A and for fumanism B1. Unlikely, we didn't find any significant reduction for the larenon and its metabolites for the AUC, but we found a statistically significant reduction for the maximal urine concentration comparing control group and bioorganoclay treated group. So we can say that this new product was able to reduce the urinary excretion of mycotoxins in these animals. Concerning piglets trials, these trials were done in collaboration with the Ghent University. We used 16 piglets sexually divided, and we provided them a single intragastric bolus of a mycotoxin cocktail with or without the new bioorganoclay. In this case, we collected blood samples at different time points, and we analyzed the amount of this sample using a LC-MS-MS method and following again the biomarker approach. Also in this case, these toxicokinetic studies provided us some important toxicokinetic parameters, such as area under the curve and maximal plasma concentration. What we can say about results in piglets? We found again a reduction for all mycotoxins tested in these trials. In particular, we found a statistically significant difference for the AUC curve comparing control group and treated group for aflatoxin B1 and for ochratoxin A. Unlikely, we didn't find any statistically significant difference for fumonism B1 and zearalenone glucuronide, that was the biomarker of zearalenone in piglet blood. But anyway, also for these two toxins, we found a trend reduction. So we can say that this product was able to reduce the bioavailability of mycotoxins tested here in piglets' blood. After that, we wanted to study some specific absorption on the new bioorganoclay. So we studied the micronutrients absorption on this new product. In particular, we used vitamin A, vitamin B12, and calcium as example of fat-soluble, water-soluble vitamins and minerals. So we evaluated the mean serum concentration of these 3 compounds in 6 piglets. And no significant reduction of these 3 compounds in serum after 0.5 and 4-hour post-bioorganoclay administration was found. Okay, in this slide, I want to summarize the results obtained in all studies. And we can say that to use a new product in vivo, of course, we need before that this product is very, very good in vitro. Indeed, we found more than 80% of mycotoxin reduction in all cases. And we can say also that despite the difference between experimental designs between rats and piglets model, results were in line for each mycotoxin tested. So concluding my presentation, we can say that we develop and produce a new bioorganoclay acting as multi-mycotoxin adsorbent. This new adsorbent was tested in vitro at different pH values, simulating those encountered in the GI tract of animals, and was characterized by equilibrium isotopes. The efficacy of this new bioorganoclay was confirmed in vivo by toxicokinetic studies with rats and piglets using the biomarker approach. A patenting of this new product is in progress. So I want to thank all my colleagues, in particular Laviosa Chimica Mineraria, where I spent 1 month of my PhD and where we developed and produced the new product that we tested. I want to thank Ghent University, where I spent 3 months of my PhD and for the collaboration in the piglets trials, and the University of Bari for the collaboration in the rats trials. Of course, I want to thank my institute, my research team, Giuseppina Vantaggiato and Donato Greco, and my director, Antonio Lo Greco. Of course, thanks for your attention.
Thank you very much, Vito. Excellent presentation. There is a question.
Yes, there are 2 questions. The one from Evelina Parisi. Which was the competition of a pH 3 and pH 7 solution, respectively?
Sorry, you can repeat, please, Alessandra.
Which was the competition of pH 3 and pH 7 solution, respectively? Is it clear?
The competition. I can say, in this case, I can say that...
There was a typo, a composition. So which was the composition of pH 3 and pH 7 solution?
Okay, the composition. Okay, for the pH 3 we used a citrate buffer, and for pH 7 we use a phosphate buffer in our in vitro trials.
Okay, there is another question from Monica Fresolone. The results observed on animals can be valid also for deoxynivalenol?
This is a very interesting question. Thank you for the question. Concerning the deoxynivalenol, we of course we didn't try in vivo trials because we tried different trials in vitro, and we didn't observe any adsorption from this bioorganoclay. So for this reason, we didn't go on in vivo for these mycotoxins.
Okay. Thank you very much, Vito. Now we move to the next speaker. That is Yukai Liao from China. His talk is titled Detoxification of deoxynivalenol in wheat by bacteria and enzymes.
Hello, can you hear me?
Yes, we can hear you.
Okay, can you see my PPT?
No, we don't see your video, neither the screen. Can you try to... Okay, now we see the screen.
Oh,
okay, excellent.
It's okay now?
Yeah.
Okay, that's fine.
Oops.
Okay.
You
move it. Start now?
You move something again because there is no video, no screen, not screen sharing. Can you try again? You did before, but now.
Is that okay now?
Yeah, perfect. Okay.
Okay, that's fine. That's fine. Thank you. So now I started. Yeah. So I think thank you for your organize this conference, and then it's very nice to have a chance to share some progress made in our laboratories. So I would like to talk about the detoxification of deoxynivalenol in wheat by bacteria and enzymes. So this is belong to the work package 6. task 6.3. So our objectives were to select the microorganisms and enzymes for biodegradation of mycotoxin contaminated feeds and foods. So our target mycotoxin is DON, the deoxyribonucleic. The main progress made in our university as detoxification of DON in wheat by 2 bacterial strains, and also detoxification of DON in wheat by recombinant enzymes. So we work on detoxification of DON by targeting the C3 hydroxyl group. So this is the DON, and this C3 hydroxyl group was found to be the major determinant of toxicity, and also that they have proposed that two steps are involved. The DON first by oxidation to produce 3-ketodon, and the second reduction to produce 3-epidon. But as before we started, there's no proof, and also there's no gene isolated and no enzymes. So this is a process called DON epimerization. The product for the 3-ketodon and the 3-epidon, they have a very clearly reduced toxicity. For example, for 3-ketodon, they have a 10 times lower toxicity compared to DON, and for 3-epidon, they have a more than 1,000 times fold reduction of toxicity. The first part is detoxification of DON in wheat by 2 bacterial strains. We isolate one bacteria is called S34. It belongs to Sphingomonas. It's a bacteria from soil, and from this result we can see that after 72 hours, just don't completely remove it, and that's for the 3-epidon. That's the degradation activity is reduced, and this bacteria, when incubated with the DON and the 3-DON, so that's growth that's almost constant, no changes. We then detect this metabolites derived from DON, for example, by HPLC. We detect first compound, compound A, and then compound B. And then when we see that at the time course, the first compound produced that after maybe a few hours, and then up to reach almost a maximum level about 60 hours, and then the second compound come out after 72 hours, and then we collect these 2 compounds and characterize them by GC-MS analysis. For example, this is the DON, the molecular size after derivatization, and this is the first compound, compound A. Actually, the 3-ketodon that has a molecular size as expected, and this second compound, the compound B, this is 3-epidon, so that's a molecular size as expected. We also analyze these 2 compounds by NMR, and this is the compound A that is proved by NMR analysis. This is 3-ketodon, and this second one is 3-epidon. We then analyze this sequence by partial sequence of the 16S rDNA sequence, and then make a phylogenetic analysis. And then this is the place for this strain. That's a Sphingomonas strain. So which is in this group. For detoxification of DON in wheat, the wheat is collected from the field, and after incubation for 72 hours, so that's a complete remove of DON by these bacterial strains. So just control. There's almost no significant differences after the same time of incubation. The second bacterial strain we isolated is Diversius species D69. So this strain has a very high activity of detoxification. So after 2 minutes with the OD, with the cells with the OD value of 2, so that's a complete remove of DON. and with lower audio cell density, just 20 minutes, and with very low, that's for 120 minutes, that means 12 hours. So we can detect there's two compounds. One is 3-ketodone, and then the second one is 3-epidone. When we see the time course, so the first compound is coming out just in 1 minute, 1 to 2 minutes. Actually, when we take out the samples, and 1 minute we can detect. That's the first compound, 3-ketodone. We also analyze by GCMS analysis. And the second compound come out a little later, just after a few minutes, the 3-epidone. We use this bacteria for detoxification of mycotoxin in wheat grains. This wheat grains also from naturally infected wheat from field. So after 3 hours incubation, just complete removal of dung, contaminated wheat. So that in control after 3 hours, there are almost no changes. So this detoxification activity is much higher than the first one, so the 3-4, S3-4. We also, based on the 16S rDNA sequence, that this confirm this bacteria is a divorces species. We then isolate the genes from those bacteria and to get recombinant enzymes to test their detoxification activities. From the first strain, S3-4, we first use a comparative genomic analysis that this R strain and this D17 strain also is detoxification strains. We found there's common genes involved, and then the last strain, that S26, that is has no activity for this detoxification by targeting C3 hydroxyl group. And we found unique genes, the 188. By this functional classification, we found the energy production and conversion, because those activity by conversion from dung to 3-ketodone, we suppose that need this enzyme. There are 11 genes in these classes. We analyze those genes. So from this 11 genes, we say, so this is a predictor oxidoreductase may be involved for this detoxification. And we indicate here this for these genes, we identify later by other methods. So this is also lots of oxidoreductase, there's different reductase. We at the same time make a back library that from this back library after selection, and then we get one clone, the one positive clone. This clone is 148 kb and contain 150 genes. There's 20 categories of genes in this back clones. For energy production and conversion, there are 6 genes. And when we first analyze those 6 genes, so there's also one gene that we identified before from this comparative genomic analysis. That's the same gene. This gene has 1,032 base pairs. That's predicted oxidoreductase is also called aldolketoreductase, AKR. We express these 6 proteins, and all these proteins showed that the detoxification activity. And then we first analyze these genes. Actually, they belong to the AKR superfamily. This is a new member of the AKR superfamily. So based on this criteria, so this protein has less 40% amino acid sequence similarity compared members from this superfamily. We also disrupt this gene from this strain genome. After disruption, you can say that these strains without this we call AKR 18A1 genes, there's no activity. We express this bacteria and purify the recombinant protein from bacteria. So there's a pure protein on SDS page, and this pure protein showed the detoxification activity that it is able to convert DONG to 3-ketodone. This is the first proof that for the 3-epimerization pathway, that it involves 2 steps. The first step is reductase, that first step is oxidase, and for this first AKR18A1 genes, you can convert reversible reactions from DONG to 3-ketodone, and then if we change the conditions, so this also the 3-ketodone can convert back, can convert 3-ketodone back to DONG. For the second cloning and expression of the genes that will get from this D63, D69 strains, this has with a very high detoxification activity. We also use comparative genome analysis. From this comparison, we found common genes from both strains. These 2 strains have both activity, convert DONG into 3-ketodone and 3-epidone. And then we compare with a lot of strains that has no detoxification activity, that from this comparison we found more than 600 genes that are unique for this detoxification characters. From this first comparison and also classification, we found there's 43 genes that belong to dehydroxylase that are supposed to be able to convert DONG into 3-ketodone. And for this second comparison that we found, for the second classification, that more than 100 genes belongs to this reductase. Those genes are supposed to be able to convert 3-ketodone to 3-epidone. And after this analysis, we also make expression analysis of selected dehydroxylase and reductase from these strains. From this expression analysis, we found that after 5 and 30 minutes after the addition of DONG, there are 9 genes has a high level of expression. That means they have a high level of transcripts. For this reductase, we also found that 9 genes have a high level of activities. From this analysis, and combined with this comparative genomic analysis, we make a bacterial expression, and then we found that there are 3 genes. One gene is quinol-dependent DONG, dehydroxylase hydrolase, that we call QDDH, and has activity to convert DONG into 3-ketodone. We also found that the 2 genes that belong to AKR gene family. One is AKR13B2, another one is AKR6D2. So that's a new sub-member of the AKR family. This is a new sub-member of the AKR family. By bacterial expression, we can see that they showed a detoxification activity after the purification from bacteria. That's a QDDH, and it can convert DONG into 3-ketodone. So there's no reversible activity. So this is a different pH and the temperature. We can see that there's this pH, there's a higher activity, and also there's a 40-degree, this protein has activity. For AKR13B recombinant protein, that also showed that activity convert 3-ketodone to 3-epidone, and this is also AKR6D1 for showed from 3-ketodone to 3-epidone, but it showed a reversible activity. And then these 3 enzymes put together, and this DONG can be converted to 3-ketodone and 3-epidone. We also put the 3 enzymes together with the weight that contaminated with mycotoxin. So after 6 hours, that's complete remove of DONG, and after 3 hours, that almost 86% of DONG was removed. We also classify mutagenesis that found that 3 amino acids are involved in this DONG and QDH interactions. And those people are involved for this work. And thank you very much for your attention. Okay, thank you.
Thank you very much. Thank you very much. Very, very interesting talk, impressive work, and there are questions.
Yeah, there is a question from Bert from Bremen. Very interesting topic. How can those bacteria enzymes be applied in practice? Is a liquid media always necessary?
Sorry, do you mean any special media?
Is a liquid media always necessary?
The
Yeah,
question
we use a liquid media for the they showed also ketogenic activity. Is that your question?
The question is if for practical application, the liquid media are necessary.
For practical application, do you mean that is a liquid media can be used for detoxification?
Well, we got it from me, so I think your first answer was fine.
Yes, sure.
Then there is a second question.
Yeah, from Michelangelo Pascale. Engineered bacteria are allowed to be used in China for detoxification purposes?
I think the first thing is that for the S34, there's low clear data for the animal experiment. For the diverse species, there are 2 or 3 papers in recent years. They have showed that with animal models, and I think the first is with the rat, and the second with piglet. They show that no toxicity is safe for use in animals. That's from another laboratory that was showed. That is for the second bacteria that diverse species that use it for additive for feed.
Okay, thank you very much for your talk, Yunkai Liao. So we now move to the next speaker, which is Michele Schumann from Italy. He will talk on the influence of processing parameters on the mitigation of deoxynivalenol during industrial baking. Michele, please.
Yes, good morning. Can you hear me?
Yes.
Good. So I've just tried to share my screen. I hope you can see it now.
Yes, it's working. It's working.
Perfect. Okay, so hello to everybody, to the different parts of the world which are connected today. I'm very delighted to illustrate to you in this Mycokey occasion event work that we performed in the parallel period time My Toolbox European founded project, again on the mitigation of toxins along the food chain, as you know, in joint activity, also in synergies with the Mycokey ones. And in particular, my speech today is dedicated to talk about the food processing parameters influence into the deoxynivalenol changes, focusing then on baking process and on bakery products. This work was been done in collaboration, in particular with the University of Vienna and IFA Turn in Boku, and we worked together with, let's say, a practical activity also in the pilot and industrial scale plants here in Bari in Italy. Let's start just with a very quick reminder about the steps that characterize the mitigation of deoxynivalenol along the food chain. You have a first cleaning and sorting step, and then the branding and milling of the durum wheat or common wheat, for instance. And in those steps, you could already have the maximum residual limits to be taken into account that could determine also and are fixed in relation to a clarified and well-established reduction that those steps can determine along the chain. The other point is related to the processing transformation of the raw materials into the end products. And there's the possibility to have a further degradation depending on the product, which is related to the formulation, but also to the processing itself. And in this sense, we have that along the story of the literature in the last, let's say, 3 decades, mainly, you could find that the deoxynivalenol changes in percentage can be, let's say, more or less than, let's say, the zero value of the raw materials, depending on different studies, because you have to consider that there's the need to find all the extractable degradation products that can be originated from DON, and also you have to consider to check the matrix and have a clear mass balance for all the destructible metabolites. In this sense, a final consideration is that when you have a change of deoxynivalenol, it doesn't automatically mean that it is a detoxification. You need to identify also the toxifying aspects related to the metabolites. And in the literature, as said, there are some compounds that are indicated as the most reported during the baking or thermal, let's say, effects on processing. And what is missing is that we still need to quantify all the degraded product form and also establish the toxicity, as said. And the literature research is not clear when you see, for instance, into the 3 main categories of bakery products, which also, let's say, involve different kind of processing steps that move from toasting to, let's say, fermentation, baking, etc. You have that moving from crackers to biscuits, and in particular to bread, the literature can completely indicate totally different results, even if more of them, when you have more thermal effects, are on reduction of the deoxynivalenol itself. And the literature report also not completely clear information about the analytical validation of the method, the kind of pilot or lab scale trials, and also the condition about the dilutions and the calculation that also take into account simply of the humidity of the different products up to the final finished one. So we decided to take into account of all the potential molecules that represent the change of the deoxynivalenol during the bakery process. And we wanted in this study to target, let's say, the complete mass balance of the, let's say, all the forms that can be extrapolated from a non-targeted approaches. We developed an analytical methodology that was based also to, let's say, real products. So we worked not in, let's say, in lab or in the kitchen, but in pilot plant scales. And we prepared the 3 different categories of crackers, biscuits, and breads, mimicking all the industrial scale trials and changing also the different parameters in the recipe. And we identify all the transformation products through an approach, on a targeted approach by liquid chromatography, high resolution mass spectrometry, and determined all the deoxynivalenol degradation during the real life transformation of the raw materials into the industrial finished products. In particular, the work has been done because we exploited naturally contaminated flour and we added a labeled C13 deoxynivalenol into the dough, following the changes of all the compounds with this non-targeted high-resolution mass spec approach of both the pairs of the not labeled and the labeled one, in order to identify and compare all the changes of the molecules. You have here an example of this untargeted identification, and you can see, for instance, that with respect to the molecule of deoxynivalenol, during the 3 different bakery processes, we identified only 3 final metabolites, and only these 3 ones: isodeoxynivalenol, Nordon B, and Nordon C, with extremely high presence of isodon with respect to all the other 2 forms. So Nordon A, for instance, and DON 1 that are reported in literature, for what concern these 3 bakery processes at industrial scale, they were not detected. we quantified the 3 different compounds into the products, and we identified also in the baking process that during different processes you can see that the reduction of deoxynivalenol and the contemporaneous appearance of the 3 other compounds is relatively reduced for bread. It has a different impact for what concerns the biscuits, and also a more, let's say, strong one for what concerns the crackers. The final point was also to take into account about the toxicity, in particular of isodone, because Nordon B and Nordon C were already tested in toxicity terms for the scientific literature, and we found that isodone, which is present in prevalence, low in prevalence with respect to the other 2 forms, was considerably less toxic, and I will show you. Because when we tested the cell viability comparing the deoxynivalenol in vitro result with respect to the isodone one, we found from repeated trials that the toxicity of isodone is from 1 to close to 2 order of magnitudes lower than respect to the parent compounds. So we can also say at the end that there is a real detoxification of the process itself due to this change of the molecule. We quantified with a mass balance the 3 different molecules found with respect to the parent one before and after the 3 bakery processes, and we identified substantially with different replicates the changes of the 3 compounds with respect to the deoxynivalenol. And I would like to focus your attention in particular to the fact that we went also in more details trying to create a real design of experiment approach, changing into the pilot plants the main technological parameters that represent the 3 processes. So baking condition, the pH of the dough, the raising agents, and also temperature, time of baking, or let's say having the concentration different from the leavening agents. So each factor was modified, but according to a technological reasonable range that could came to, let's say, a finally acceptable organoleptic products. And we also extremized some trials, as you can see from the photos, to model and create a predictive model from this design of experiments approach. And the predictive model at the end of all the trials returned to us an information about, let's say, the different influence of the main parameters into the changes of deoxynivalenol into mainly isodone, and then in a second time Nordon A, Nordon B, and Nordon C. And you can see, for instance, that for what concerns the biscuit process, you have that the most relevant parameter that influence the change is represented from the pH of the dough through the raising agent, in this case sodium bicarbonate. The other aspect that you consider is the fact that the other parameters are relatively not influencing so much the process. If you consider the cracker one, then you have that together with, again, the first most important influence represented by the pH, you have the baking time as the second really influencing parameters in terms of applicating and mitigation effects into the final cracker industrial product. So the outcomes of this study are represented by the fact that considering that deoxynivalenol has been reported as to be unstable in alkaline solution, the pH of the dough is really an important processing factor at the industrial scale that regulate the transformation into the isodone one. And we found also that with respect to, let's say, the degradation of deoxynivalenol, then you have to consider the fact that increasing the presence of the raising agent, you can explain the fact that also in the 3 different processes you have a different effect, which is not so much relevant for what concern the bread, in which also you have a natural fermentation. For what concerns the baking condition, the real influence is only practically in the crackers process. And this could be explained taking into account that you have a completely different moisture and a completely different surface to volume ratio of the cracker products with respect to biscuits. So the kind of thermal impact that you can have during the process is really different if you compare the two products, the crackers and the biscuits. And this can explain why there is a significant influence also of the baking time, as we have seen, together with the pH of the dough into the final transformation of deoxynivalenol into the less toxicological relevant forms during the process. So at the end, the take-home messages that we can identify is that for the first time, we really have a complete picture of all the compounds that originate from deoxynivalenol during the bakery processes that we were able to classify through untargeted stable isotopically labeling assisted by the liquid resolution mass spectrometry. And in addition, now we found that the main transformed compound, which is represented by isodone, is considerably less toxic. So this proves that a large-scale industrial bakery process can really result in a partial detoxification of deoxynivalenol, which means that beside to all the other actions that we can do along the chain, also, let's say, the real optimization of some parameters within the bakery process can improve the safety of this product in relation to the final consumers, because this could be, let's say, a further action that can complete the entire work that along the chain can be done in order to mitigate the risks. And with this, I leave you with a couple of indications about the recent publication of last year, which reports all the results that I illustrate to you today. And I would like to thank all my toolbox partners, in particular the one here reported that collaborates directly to this food processing activity, and all of you for your kind attention.
Thank you very much, Michele, for the very nice, quick, and clear presentation. There is a question from the audience.
Yes, there is a question from Christine Wings. Were enzymes used during baking?
Did you listen to the question, Michele? Michele is not online anymore. Ah, what a pity. Michele? He has lost the connection. Okay, sorry, we were Michele, can you hear us? No. Okay, Michele, we thank you again, and maybe we will forward to you by chat this question, and you can reply to the participant that has posed the question. Okay, next speaker is now Paola Battilani. We open the session Modeling and ICT Solutions. Paola Battilani is a work package leader of the work package 1 global mycotoxin knowledge and in Mycokey project, and together with this big duty that she had, she has also got the time to work on the modeling during this project in work package 2. So a lot of work for Paola. She will talk about modeling as useful tool for mycotoxin management in the field. Paola, you should activate your microphone, please.
Can you hear me now?
Now perfectly. You can go.
Okay. Thank you very much, Antonio, for the nice introduction. And I'm really happy to talk about modeling because, as everybody knows, it is a very important topic in my research. So modeling is, in principle, a simplified representation of reality. Therefore, to simplify reality, we need to find out the main factors that influence the story we are trying to tell. In this case, mycotoxin contamination in different food crops. This picture is just to highlight that we are working with several fungi, several crops, and the main source of variability in the fungal population is surely the ecology of fungi. This slide regards the different temperature level, and we can see that depending on the fungus, we can have different optimal, suitable, or unsuitable conditions. The result of this important impact of ecology on fungi is very well highlighted in this geographic risk map that is the output, one of the output of work package 2. But we also must keep in mind that each step of the infection cycle of fungi in relation to the host crop can determine a change in the behavior and therefore in fungi and mycotoxin that we can find on our final product. For example, talking about maize, we have different fungi that can be found on the crop, and each one has a different need, for example, for survival, for dispersal, or also for growth and mycotoxin production in the grain. So why I introduced this very important impact of fungal ecology? Because in principle, the weather we have in the growing area of the crops is the main leading variable that can be used for modeling. So modeling, I will focus on predictive modeling that are considered the best approach to have a suitable prediction that can be easily adapted in different geographic areas. So starting from weather data, when we approach modeling, we can use those data to predict crop phenology and also to predict fungal infection cycle. So combining crop phenology and fungi infection cycle, we can have a prediction of what happens regarding the risk of contamination of mycotoxin in the final product. In this case, I'm talking about maize, so we can have a prediction of what is happening in the grain and which kind of risk we have for regarding different mycotoxin contamination. This is just an example. We have here three different pictures that represent a daily dynamic prediction. In the first figure, we have a low risk. In the center, we have a medium risk, and in the last one, we have a high risk, which means that during the growing season, starting from the crop is susceptible, on a daily basis, we can have a prediction. So we have, we can be prepared regarding the final contamination of our crop. So how can we use this information? Using actual meteorological data, day by day, we can give an information regarding the ongoing risk to farmers. But this is also very interesting for extension services or for all the stakeholders that are involved in the chain, in the maize chain production in this case, but obviously for all the crop production chain. But I think it is very important to keep in mind that we can also use other kind of meteorological data as input for our predictive model. For example, if we use historic data, I mean, we have data coming from the last 20 years, we can run the models with the historic data and we can generate past scenarios. I mean, if we consider a geographic area, in that area, we have each year a level of risk computed for the whole area, and taking into account an historical series, we can draw a risk map of the area. Combining the risk map of the area, which is an historic information, and the actual information that comes from the weather data collected on a daily basis, we can surely support farmers. First of all, we have a focus on risk areas, which means in case I am in a high risk area, I can apply different cropping system or different harvest time or methods, and also crop harvested product management to minimize the mycotoxin contamination. Another very important point is that we need to sample and analyze a lot of our products because of the legislation in force. But when we have a knowledge regarding the risk areas, we can optimize sampling and analytics, focusing mainly in the areas where we have a high risk. So I think this is a very good support for farmers, extension service, and stakeholders. But I just want to show you how it can be used in practice. For example, selected a geographic area, we can have several weather stations. You can see several points on the map. For each weather station, we can deliver on a daily basis or on a weekly basis a prediction of the risk, and the farmers and all the stakeholders can take into account the prediction. In addition, we commonly combine the date of prediction and the level of risk of the area and the prediction of the model, and we deliver short messages to farmers. This is just an example. When the probability of high contamination is above 70%, some suggestions are addressed to the farmers and the stakeholder. This is just an example, but obviously for each combination of risk area and prediction of the model, we have a different suggestion for farmers. There is a further possibility to use predictive model, which is using future data. And this is really very important, actually, because we are living in a climate change, and future is absolutely unknown if we don't try to predict based on modeling. The maps you can see here comes from a project aimed to understand how aflatoxin contamination can become relevant for Europe. And you can see that depending on the actual +2 degrees centigrade or +5 degrees centigrade scenario, the situation is completely different. And the color immediately show us that when the color is red, we have high risk. So it is very clear that in Europe, we are facing, especially in the +2 degrees centigrade scenario, an increase in the risk of aflatoxin contamination in maize. Therefore, this kind of approach is useful because, again, risk areas are highlighted, and based on the future prediction, strategies can be defined by decision maker, institution, but also researchers can take into account this prediction and address their research mainly on the issues that will be more relevant in the future. But let me say that climate change is not only a problem that a policymaker or institution must face, because it is also something that farmers face almost every day. In fact, models, predictive models are useful, and they are relevant for stakeholders when they are accurate and reliable, which means when we have a predictive model, we need to validate the model and define the level of accuracy we can reach with the kind of predictive model. Because if we give to farmers, to stakeholders, wrong prediction, obviously they are totally unhappy and they stop following our prediction. Unfortunately, the accuracy of prediction in the last years decreased, and it is mainly supposed to be related to climate change. Further, the impact of the cropping system increased during the last years, and this is I think it is justified because during the last 20 years, we devoted a lot of efforts to define guidelines for farmers and also for all the operators of the crop chains to improve the cropping and management system to reduce mycotoxin contamination. Therefore, these 2 points, climate change and guidelines for crop management, requested an improvement and update of predictive model. Just a reminder, climate change is something that is known to everybody. We are facing a higher temperature, higher or different rain distribution. But first of all, we are facing extreme events, and the frequency of extreme events is increasing a lot. So when we have extreme events, any prediction becomes much more difficult. And what we noticed recently is that we can have in the same geographic area, even in close places, a very different level of contamination, and especially the co-occurrence of different mycotoxins. In this case, I just wanted to focus the attention on the color of the bars. The numbers are 9 different geographic areas, but they are very close to each other, maximum 130 kilometers distance. And you can see that the green bar or the blue bar that represent fumonisin and aflatoxin are very different in the different geographic area that I underline are very close to each other. So climate change is considered responsible for this kind of co-occurrence and very variable contamination in maize crops that are geographically very close. But if we go to this graph represents the aridity index, which is a way to summarize the meteorological data collected during the maize growing season in the 9 points I show in the previous graph. As you can see, we have different bars, different errors, which means we have a very, very wide variability in meteorological conditions. And this explains why we have a very different contamination, both in terms of amount and also in terms of co-occurrence of different mycotoxins. So this point is crucial for modeling because till now, predictive modeling considered each fungus singularly. I mean, there is a model that is able to predict aflatoxin contamination in grain, another one for fumonisin contamination in grain, but it is no more possible to consider separately fungi, because in these very variable weather conditions during the growing season, the co-occurrence of fungi on the host crop is a reality. Therefore, in Mycokey, we approached the impact of co-occurrence of fungi on mycotoxin production. It was done both in vitro trials that I show in this, summarized in this slide, and the colored area represent the growth of Aspergillus flavus and Fusarium verticilioides in different temperature conditions. So you can see that when fungi co-occur, there is a different behavior. Apparently, there is mainly a reduction of the fungal growth, which is more relevant when Aspergillus flavus influence is considered on Fusarium verticilioides. But I think much more interesting is the impact on mycotoxin production. Because again, we can observe a reduction, but for aflatoxin, we also see a switch. The optimal temperature for aflatoxin production seems modified. It is no more 25 degrees centigrade, but it is apparently 20 degrees centigrade, which is apparently a nonsense. But moving to the field results, we inoculated singularly or in co-occurrence the Fusarium verticilioides and Aspergillus flavus. And here you can see the production of fumonisins and aflatoxins in two different years, 2016 and 2017, when fungi were inoculated singularly, the light gray bars, and in co-occurrence, the black bar. The 2 years showed a completely different behavior of regarding mycotoxin production. In 2016, in the co-occurrence, we had an increase of aflatoxin contamination compared to the inoculation of a single fungus, while in 2017, the opposite happened, and more or less the same happened for fumonisins. What can we say regarding the 2 years? During the ripening period, mean temperature in 2016 was 23 degrees centigrade, while it was much higher in 2017. So let me say that apparently the field data and in vitro data are not in agreement, but if we think about the final information I gave you, we can find a reason because we have a different level of contamination. So the result of these trials is the basis for an improvement in predictive modeling. In particular, we developed a joint model that consider not only the weather information as input, but also the interaction between fungi. The results, the validation of the model regarding the fumonisin, which is on the right, was good because the correct prediction were high and increased respect to the previous model, while for aflatoxin it is not absolutely it is absolutely unsatisfactory, because as you can see, the correct and the wrong prediction are more or less the same, which is absolutely not a good result. But let me say, we are still working, because it is very, very difficult to include in a predictive model the strange behavior we observed in field and in vitro. So we are still working for improving aflatoxin modeling. The last point regards the crop management. We have different action we can do during the cropping system to try to reduce the level of contamination in grain. Therefore, it is important to include also this information in predictive modeling. But till now, nobody succeeded in including, in defining mathematical function able to take into account the cropping system. And this is not surprising, because we have obviously not a single action in the cropping system. We have different actions, and all action we do interact with all the others. For example, if we select an hybrid, the susceptibility is different. If we apply irrigation, it interacts with the hybrids. If we, I don't know, change the manuring, we can have another interaction. So till now, cropping system was never included in mechanistic models, because it is almost impossible to find a mathematical function to describe what is happening. So a step forward was necessary, and in particular the machine learning approach was used. And this is more or less the summary of results. As you can see, when we consider the cropping system, which is the graph on the right, compared to the left, we have an improvement in correct prediction, both for aflatoxin and for fumonisin. And because we have several years considered in this study, let me say that more than 75% of correct prediction is really a good result for a modeling approach. So models are the core of IPM, because based on the European legislation, IPM takes into account all the data collected, but to give information to farmers, models are the core. But they are also the core of ICT solutions, because ICT solutions are based on modeling. Therefore, we should expect to have a big effort in developing predictive models for mycotoxins. But let me say, this is not the case, because if we take into account all the paper published on mycotoxins, we have more or less 10% of research based on model, but not predictive model in the crops, but just in vitro models. and mechanistic models available are very few. I focused on maize because we have two predictive models for fumonisin and aflatoxin in maize, one prototype for deoxynivalenol in maize. Most of efforts were devoted to fusariumab blight on wheat, and recent researchers focused on aflatoxin in pistachio and ochratoxin in grapes. But let me say that for the last two models, the validation is limited for pistachio and not yet available for grapes. So I think most efforts should be devoted to predictive modeling in view of the role predictive model can have also in the next future. So just to conclude, I would like to tell, to summarize the story of modeling in the EU context. The first project on mycotoxins I was involved in was the cost action. Then we had the mycotoxin cluster in the 5th framework program, where the attention started to be focused on fungal ecology and fungi characterization, also molecular characterization, and basic modeling in vitro, taking into account one variable each time, started. Then there was an improvement because mycotoxin became more relevant for farmers, and the cropping system was considered, so as climate change. Then we moved to a further step, and predictive modeling was included in MycoRed. But it is also included, obviously, because I'm telling this story, in Mycokey, and fungi occurrence and the impact of cropping system and ECT solution were inside the project. But now I only showed you that using the co-occurrence of fungi, we can improve models, or we are trying to improve models. Considering the cropping system, we can further improve models, but we have now many other challenges because, for example, gene expression, the connection of modeling with the remote sensing, and many other topics can be related to included with modeling and give a further robustness to prediction and a more reliable support to farmers. We hope, therefore, to have a future project that can take into account these further improvements. That's all. I would like to thank the Mycokey project, but also other projects that were in connection with our work, and especially Marco Camardo and Paola Giorni, that were my main support in this research action. And obviously, I thank you for attention and the organizer for this very interesting Congress.
Thank you very much, Paola. Very great overview about the modeling, and let's start to work to write the proposal. The topic of the points that you have raised in the last slides are extremely interesting, so we must soon be in connection for writing a nice proposal. There are questions, and I have also a question that was made in the first day, but to Chris, and that I will forward to you later.
Yeah,
so Alessandra, please.
Sure. There are three questions. Michelangelo Pascale asks, climate change and mycotoxins risk maps based on data. Can you hear me?
Sorry, I don't.
Yeah.
There was a bad.
Yes, I will.
Can you repeat the question, please?
Yes. Okay. Can you hear me now?
Yeah.
Okay. Climate change and mycotoxins and risk maps based on data occurrence. I think that harmonized collection data should be necessary. Same sampling, protocol, location, agricultural practices. What is your opinion?
I think this is a crucial point. In fact, when I mentioned that a couple of predictive models were not validated, the point is exactly this one. We don't have an organized data collection, and to have reliable maps and reliable predictions, we need to harmonize data collection. Keep in mind that especially for modeling, but also for risk maps, we have a geographic base. So we need the geographic coordinates of the sampling points, so as meteorological data, and it is crucial to have an harmonization, otherwise we cannot reach a good result for a wide community like the European community.
Okay, there is a second question from Susan Vogelgang. Maybe I missed it, but what are the cropping factors in maize increasing or decreasing the risk in apple production?
As I commented, based on the machine learning approach, it is not so easy to find out to discuss on a single point, because we have an interaction between different action on the crop. So I can say that irrigation is very important for aflatoxin, and this is known. Or the seeding period, the sowing period is important both for aflatoxin and fumonisin. But if we try to give a value to this impact, it is quite difficult. While with the machine learning, all the interactions between all the factors are taken into account, and therefore in this way we succeeded in improving model performances. So I can list the main relevant actions, but to have a quantitative evaluation of the impact, it is important to take into account in the same time all the cropping system, the whole cropping system applied.
Okay, thank you. There is another two questions. So one from Claudio Altomare about the co-occurrence of mycotoxigenic fungi. You studied the interaction through an in vitro model based on co-inoculation of fungi in a kind of confrontation test. This condition implies a competition between strains for limited space and nutrients, which is not what necessarily occurs in vivo in the field. How can the results of such a model be used to infer future scenarios?
I totally agree on the comment, and obviously the point is that we can only have a possibility to study in detail different level of temperature or water activity and so on, and so we managed the trials in vitro. But we, in the meantime, we did the same kind of trial in field, in a crop, and we inoculated the two fungi. Really, we used also Fusarium graminearum on the hairs. So we tried to have the true situation where we cannot change, modify the conditions, and the in vitro situation where we can change in detail each factor we want. my answer is that to translate the results in something that can be useful for modeling, we must match the information we get. And as I told before, for fumonisin, the approach is easier. For aflatoxin is much more complicated. And therefore, I don't have a true answer to the question, but surely it is mandatory to use both in vitro and in field data to try to find a support for modeling. Because I agree, in vitro we have also the competition, which is not the case in field, but no way. Otherwise, we should grow maize in chambers, but maize is very difficult. It's a big plant, and we need the grain. We need the final product to be sure that what we are doing is in agreement with the real situation in field.
And 2 more questions, and then we have to move to the next speaker.
Enrique Piuk asks, Dear Paola, how far you are from commercialization of mycotoxin prediction tool? What is your opinion, barriers and main tasks? What needs to be done?
The question is commercialization. We really, because we developed the model starting from public money, we have not in mind to use for a commercial aim models. Anyway, the models are regularly used for the Italian farmers, especially living in Emilia-Romagna region, because we have a strong interaction with the regional government. We have some collaboration. For example, on the first day, there was a talk from Biomin. We had a collaboration with Biomin because they were interested in our predictive modeling models for maize. And so they started from our models and they applied according to their need. So we established a kind of collaboration, not commercial use of models. And I think this is the best way to use our predictive models because they can be targeted with for specific aims, and the scientific results are, let me say, safe, because everybody can use the information, but not, at least till now, we never considered a commercial use. In Mycokey, anyway, we delivered the predictive model for the ICT solution, and Theo will talk after me. And this is a way to use the models. It is sure that if we want to keep away to keep active the app, possibly we can receive money from farmers or from organization, but it is not at the moment our aim. If we talk about the reliability of models, I think they must be monitored, because, for example, the decrease in reliability was observed in the last 6-7 years. So it is very important to keep the interaction between the users and the developer to try include improvements in case model performance decreases.
To this respect, I think we need a lot of data to be produced on the epidemiological aspects for keeping the models I suppose. Next question.
Yeah, last question. Ahmed asked, in your opinion, do the modeling is effective in an area like the southern Mediterranean basin, where the weather seems unstable from year to other?
Sorry, can you repeat the question?
In your opinion, do the modeling is effective in an area like southern Mediterranean basin, where the weather seems unstable from year to other? Did you?
Well, I don't understand the last part of the question.
I mean, it says the weather in the basin area, in the Mediterranean area, is unstable from year to other. Change, no, where the weather change. So he's asking in this area the modeling could be effective.
Yes, I think so, because the approach I showed you is based on mechanistic models, and they perform in a comparable way everywhere. If the point is, are the farmers in southern Mediterranean basin ready to accept a suggestion from models? Really, I don't know, but next Monday it will be organized a meeting for African people regarding modeling. So I think the interest towards predictive models is increasing, and I think it should be also more interesting for those countries to have predictive models because they have less facilities and less they are less aware regarding the problems related to mycotoxins. So with a predictive system, I think the African countries, for example, can depict a kind of risk areas for each country, and they could focus on where the problem is more severe each year. Then we have a lot of other things to discuss, because the crop management in some countries are still very far from our level of attention, and so modeling is not a solution per se, but I think it could be a very good support for also for farmers in any geographic area. We also had recently collaboration with partners from Africa, because they wanted to have the application of our models with their meteorological data, and the results were interesting for them. So I think there is a lot of space for interaction, and a good way to support farmers in any geographic area.
Okay, thank you very much, Paola, for the long discussion after the talk. Maybe we were too long, sorry, with the audience. And the next speaker will be Theo van der Lee from Netherlands. But before, we would like just to give 30-second words to Michele Schumann that wanted to just say hello, because it was he lost the connection. Michele, are you there?
Yeah, I'm there. Can you hear me?
Yes.
That's good. Okay, thank you. Yes, just because when we stopped the presentation from the full screen, all the line fall down, so I was not able to say hello and take my regards to the people, and also answering to the question about the enzymes. So quickly, very quickly, just to answer to the fact that no enzymes were used directly into the biscuits, bread, or crackers processes I showed. Only mother dose was applied for the cracker process, but from the design of experiments, it demonstrated to be a low influencing parameter with respect to the others. So just to give answer to the question from the audience and say hello again from me.
Thank you very much. Very kind of you, Michele, as always. And then the words to Theo van der Lee, the work package leader of the ICT solution. He will talk about Mycokey app, an ICT solution to facilitate mitigation of mycotoxin risks. Theo.
Okay, thank you, Antonio. So it's my pleasure to present some of the results that we have obtained in this project. And I will see if I can share my screen. Can you see my screen?
Yes, we can.
Okay. So now it looks like, yes. Hopefully you can see the next slide now. So agriculture is really changing, and many people already mentioned this during this conference. So I think there's three major things that are changing at the moment. The first one is that there's new cloud computing services, new devices, and that will generate a lot of data. At the moment, we're still lacking data, and that is also mentioned at the moment. We still need more and more data to come in, but this is going to change very rapidly, and some of the partners already showed how that can change over time, and also what is the need for that. The second thing is that there's image and map overlays that create an augmented reality. So that expands actually our control. So what we can see by eye, that is not always what we need to know. So we can try to identify other sensors, other possibilities to have overlays, let's say, on the data in the field. And that can help us to make better adjustments and better predictions. And then finally, there's also this Internet of Things. So there's near field communication, and that provides a lot of possibilities to connect, not between humans per se, but also with equipments and different computers and programs. So in the end, that will become possible, I think, also, and can be very important in agriculture. So below I saw a few slides. So you think this is the advancement of agriculture over the years, but in fact all these pictures are quite recent, so you can see a whole range of things happening at the moment, and we also have to realize that not everyone is in the same position at the moment. But what we would like to do is to empower farmers to make use of all these new facilities, and one of the things that we work in Wageningen is that we try to get all the data from their own fields back to the farmers so that they can manage their crops more efficiently. So this is actually quite difficult, quite a challenge for them, because it's difficult to collect and integrate these data sets. But there's platforms that can do this now. And then the key thing of this is to have a geographic information system. Paolo also mentioned this, so it's important to locate every step and to make sure that you have the information located on a specific spot. So within Work Package 7, we had several objectives and tasks. So the milestones and the deliverables that are all mapped in this project, and we'll go to it in a little bit more detail in the next slides. So Paolo already mentioned it. I think it's quite appropriate to do the modeling and to predict things. There's a huge power of prediction, as you can see on the picture. So if you know if it's going to rain, you can take your precautions and make sure that you can move smoothly even in these adverse conditions. So it is very, very important, and more and more people depend on these predictions. In the Netherlands, we have a lot of weather variations, so we are already quite used to using this on a daily basis to check when we are going to go shopping and things like that. But this is also for farmers, this is an important thing. But there are some challenges, and many people already mentioned this. So David Miller mentioned some challenges with regard to climate change and different cropping systems. Zhang Hao and Mei-Xin Yang also mentioned that there's changes in the populations that could affect our prediction capabilities. Paolo also mentioned that this is a huge challenge, actually, with all the variables that we have at the moment. in general, the mycotoxin accumulation in different areas, and even within the different fields, can be quite substantial. So that is what we are facing. But still, we have to mitigate the risks, and we have to target the rapid tests and the combination of batches and redirect batches and clean batches. That all has to start with prediction. So the environment that we try to build on is to meet these challenges is to get data in. So we need data in the structure with the reference information from the partial information, the height and soil maps, weather and information, the rain radar, the satellite images, sensor informations. So all these things have to be integrated in this geographic information system. And if you do that, you get a lot of details. So now we're still working mainly at the plot level, but with these drones, you can zoom in even to plant level. And I think at the end, we will manage agriculture in a completely different way, in particular focusing on precision agriculture. And as I mentioned, there's other things that we can do when we have all this information logged to a geographic information system. So here we have some overlays that I showed before. So we have sensor data for home killing of the fuel uses of tractors, task maps for the tractors, and also yield maps that can be produced after harvest. And that can all be used for prediction and getting more grip on the crops. But there's more things coming. So we have new sensors, spectral images and thermosensors that can help us to get different views of crops and may help to get a better assessment of the crop's growing stage and also the harvesting time. So on the Aquabec platform that we use for the Mycokey, we have close to 6,000 accounts running. We have decision report systems from Wageningen, from Agrifirm, and Syngenta. So it's a mix actually of public institutions and also companies. We have connectivity with data service like the weather. We use it in many demo projects and also in research. And we have also partners all over the world actually that are working on this platform to collect data and to do the measurement for growers. So a little bit of technical information. It's cloud-based. There's no vendor lock-in, so you can go out anytime. It's scalable, it's serverless, and it has different connectors and front end in Angular 7 mainly, and the back end. So these are some technical things. It's an open platform, so people can use it as well. They can put up their own app in this environment, or they can retrieve data as an API to get connected. Another important thing is the data governance and security. So Aquaweb guarantees actually that the people will have their control of their private data, and that's very important for farmers. They don't want to share all the information to anyone, but they want to have control and they own the data also in this platform. It complies actually to the data code of conduct for several organizations, and growers can share data with others on the platform, for instance, for a defined period, so let's say a week or a month, and they can also have a defined purpose for that. So it's not, you don't share it to everyone for any period. No, it's a defined period and it's a defined purpose. And recipients cannot distribute the data, and the owner can also recall data, and then the data will be deleted from the recipient. So this is important to have growers in control of their own data from their own fields. So a little bit about some of the data that we used for the Mycokey app. So this is the weather data. So for instance, in the Netherlands, we have 5,500 private weather stations and 16 governmental weather stations. So the output is every 5 minutes or every hour. So that's a lot. And the reason that we have a lot is because you have to be able to curate these data sets. And therefore you need quite a good redundancy actually to be accurate in the weather. There's also weather prediction in this. So there's a 15-day interval. That's updated every 10 minutes. It's a 500 by 500 meter resolution. And the actual weather is also updated every 10 minutes and also in a 500 by 500 meter resolution. And then we have the historical weather information. And Paolo already mentioned that. I think that's very the strategic research position actually to have also historic data, because that can be used to optimize models and to check and validate models. And the historical data is for over 40 years, and that is in a 30 by 30 kilometer grid. So here we have the clean historic weather data that we have at the moment, mainly focused on Europe at the moment, but there's also sites that we monitor in South America, North America, and Africa, and China, and also in India and Australia. So this is all data sets available from specific locations, and sometimes it's for the whole country. So these are the things that are stored in this clean historic weather. So there's a lot of things that can be served as input for modeling. So the Acrobat platform is free of charge, so you can just make an own login and a user account. You get a code, and then you can start working on this platform free of charge. And then you can work on the different languages. Also, we translated it into different languages so that also people are addressed in their native tongue. So one of the apps in the Acrobat platform is the Mycokey app that you can see over here. And if you go to that app, it will know where your plots are located. And if you are following your crops already, otherwise you can add your parcels in the app as well. That's also free of charge. And then it will know which parcels are grown with particular crops. So in this case, there's winter barley, or winter wheat that is used in this particular field. And if you open your Mycokey app, it will directly show actually the current status of this crop. And in this case, it's green. That is indicating that everything is okay, and there's no risk of accumulating too much mycotoxin in this crop. So if you click on that field, you get a little bit more information. So you get this gouge indicator indicating how much of the mycotoxin is expected to be present in this situation with the conditions that you selected. Based on the actual weather, this calculation done very quickly. And you can also prepare different scenarios. For instance, in this case, what will change if I had another pre-crop? In this case, it's maize as a pre-crop, or a different cultivar that is more susceptible. In this case, the differences are not dramatic, and it's all within the limits, I would say. But in fact, sometimes it can change quite a bit. But this year in the Netherlands, the risks were quite low for the DON accumulation in the winter wheat. So there's I focus a little bit on the winter wheat, and it will also show you where it was derived from. So the calculations, the publications on how the models were derived. And so everything is accountable, and people can check these publications if they like. The transparency is a very important part. How do you come to the actions to mitigate the fusarium mycotoxin risks in the Netherlands? For instance, I have some examples. So there are several actions that you can take pre-harvest, and that is the main thing for the mycotoxin risk at harvest, obviously. So you can see the different options, and you can see the respondents in the Netherlands. Most of them actually take many of these measures. So several of them are actually done close to 100%, but in many cases they are more than 50% of the farmers take this measure to mitigate mycotoxin risks. There are two that are a little bit lower, and that is the fungicide use at the flowering, and there's the biological control. Only 20% of the farmers indicated that they are also using biological control to mitigate mycotoxin risks. If you then look at the different measures that they take, the number of measures that they take, you can see that they take mostly more than 6 different measures, and that normally results in quite a good mitigation of risks. And if you go to the what if scenario, for instance, on my toolbox, you can see that in these type of cases, the risk is very low. The weather was also quite favorable for mitigation of risk, or it had a low risk this year. If the weather would have been favorable, even then, the risk would be quite low still, if you take these precautions according to this toolbox, and also according to our calculations that were really on site for a particular grower. So if we present this type of app actually to partners and stakeholders, we get obviously questions, and many of the questions were related to the weather data. Particularly in the past, we had really difficulties to get good quality weather. I think now we have quite a good set of data for the weather, but still there's there's shadows, particularly in mountain areas, and also close to trees or things like that. You can have shadows, and for particular farmers notice that there can be huge differences in disease incidence, for instance, in these parts. And there's also altitudes, obviously. So if you have higher altitudes, yeah, that can also have effect on the weather data. And sometimes, it is it's quite steep, and it can have an effect on the weather. So on the prediction models, so the most question people ask normally, so are they trustworthy for my situation? So can I really use this? And then obviously that relates to the local conditions, and I will come to that later. So there were a lot of questions raised on what is included in the models, and Paula already addressed several of them. And it depends actually on what people are used to. And I think many of these models are actually really targeted to a particular area at the moment, and they function best in that particular area. If you go to different regions, you have different ways of cultivation, you have different possibilities to intervene, you have different strategies. So that means that actually you have to check and validate actually this model for that particular location as well. So, and the same is true for the advice. So the advice that can be quite useful for, let's say, an Italian situation may not be very useful in the Netherlands, or the other way around. So I think it's very important to connect to local partners to be able to let's say, to test and validate the models, and also to get the best advice based on these model predictions. The other thing is that we noticed that we try to keep it simple for growers, and to avoid confusion, and also to help them to make the right decision in a quick way. And the thing is that they don't, at least in the Netherlands, they don't really steer towards mycotoxin mitigation. So they're really more interested in yield, and more interested in other types of aspects than in mycotoxin mitigation, because normally it's not perceived as a very high risk, taking the measures that they already have in place. So that's why if you look at the Mycokey app at the moment, it's a Gauss indicator that can be quite useful if that is your main thing you have to respond to. But if it is another thing that you have to respond to, like for instance the growth of your crop or the yield, then actually that is the gauge that you want to look at. So in this case, you will have the performance, the crop performance as your Gauss indicator. And in fact, what people told us, the app on the mycotoxin risk, that is more like the oil pressure indicator. So normally, if nothing is wrong, you wouldn't have to worry too much about that. Only if it pops up, as a real threat, you would like to know about it and also know what to do in that case. So instead of having a special app for the mycotoxin, it would be good to have a widget that is actually integrated in an app, and I will come to that later. Because we have modified the AccuWeb platform a little bit to be able to deal with this kind of requests. So we have several apps now into these widgets. So for instance, here you have the overview dashboard of which automatically updates all these different information from these different APIs or apps, and then it will show it, in a quick view, and you can see, well, maybe there's something that you have to look at. For instance, they have an indicator here, a notification to the grower that something is wrong in this part, and he maybe has to deal with this. So if you go to the different aspects, in this case, for instance, you see the forecast for the growth and the yield of the crop. So that's very important for growers, obviously. And then you see this is the predicted yield, and there's some variation here, and this variation is partly due to a specific stress. In this case, it's a drought stress, actually, that is apparent for this crop. And in this case, an obvious way to mitigate this is to use irrigation or to spray the crop, and to make sure that this stress is relieved, and then it would also increase the yield. So it will give, you have an overview, dashboard overview. You click on the things that are of most interest at that point, and then you have your possibilities to take action. And here you have also information on the water content, for instance, on the soil. That is based on calculations from satellite information. And I think that can also be very helpful in including that type of information in the modeling. Because you have this information from a particular site, you can use it in the modeling as well. So the future perspectives, I think, for this type of apps, it's already mentioned. We have real global challenges, but I think we need local solutions as well. There's no single model, perhaps, that can do everything for all locations. It is important to at least validate that model on the specific location and make sure that growers are familiar with that, with the app, with the models, as Paolo already mentioned. So it's good to meet and to show also what would be the potential prospects for mitigation of risks. So that is something that I definitely learned during this Mycokey project. There's something that is also important in this aspect, is then to link with partners that would like to test, to compare models, and to validate mycotoxin prediction models as well. So for that, we need information, and sometimes this information is readily available. And we are in discussion with some users, potential users, that would like to use this app and to validate models in their situation. But for other regions, actually, this information is rather scarce, and it needs to be generated still. Then we're also looking for partners that would implement decision support systems, really on an on-farm scale, for instance. And that is important to talk the language of the growers and to know what are the possibilities for these growers in a particular location. And the possibilities is to use this farm map platform, as I just introduced, but it's also possible to use your own platform and to have an API link to this to the farm map platform, and to use the data that you would like to receive from that and integrate it into your own systems. So on the Mycokey, there's also the ability to to have 3 different levels of risk assessment. At the moment, we have the global, the worldwide level of risk assessment, regional levels of risk assessment. And I think that's also an important aspect. But finally, the last, and is maybe most important for growers for sure, is to have all the information integrated into an app, and be able to mitigate the different stresses, including also the mycotoxin risks in the on-farm situation. And with this, I would like to thank you for your attention, and I would like to acknowledge the people that have worked on this project. particularly Thomas Bain worked very hard on to get all the models in and up and running into the Mycokey app. We have collaboration with the University of Ghent on the modeling for the DOM in winter wheat. And Paula, she already gave a very nice presentation on the modeling in maize for aflatoxin and fumonisin. And we also had the connection with Ispa on the modeling and the management actually of this this ICT tool within the Mycokey program. And with this, I would like to end the presentation, and I'm happy to answer any questions.
Thank you very much, Theo. Very nice talk about this Mycokey app. So there are no questions from the audience, but I have a question. So how can we find the app on the App Store on the for the stakeholders? Do you think?
Yeah, so if you can go to the Akkerweb platform, and then you can register, and then there's an app store, and in the app store you can click on the Mycokey app and use it. Obviously, at the moment there's not much crops out there, but still will give you some information. And but yeah, it's freely available at the Akkerweb platform. Also available, yeah. So it's distributed in many locations already. Yep.
And as Paula said, since we have something still to do in the next call for mycotoxins, we can also include the full application of the app, the full, let's say, increase the contents of the app. So of course this is a wish for a further cooperation among the networking. Okay, I think we finish the first session, the first part of the day. Thank you very much to all speakers. We would like to meet in 30 minutes. So we will have a short delay, 13:15, for the next session that will be the voices from food and feed industries. So I'll see you in 30 minutes, and thanks again many thanks to all speakers of this very, very interesting and fruitful morning. Food and feed industries, and I like to introduce you, the first speaker, that is Jörg Ray from Serbia, of the Patent Co, and the title of his presentation will be Company Pitch and Mycotoxins Related Activity at Patent Co. So Jörg, please, you can start with your presentation. And of course, sorry, just want to thank again all sponsors and Patent Co for being a platinum sponsor of our conference. So you made possible all these two conferences. Thank you.
Thank you, sir. Thank you, Antonio. Can you see my desktop?
Sure.
Everything?
Yes, everything is fine.
Okay, I just move it. I'm just trying to see. Okay. So I don't know how this goes away. This thing.
We can see everything. It's fine.
Okay. Okay. So I just want to say thank you very much to the organizers, and thank you very much for giving us opportunity to speak at this very important conference, and particularly at these very, what you call it, strange times of the year. My name is Jog Raj. I am working as R&D manager at Patent Co. And today I will just briefly introduce our business, what we do actually as a company, and what are our R&D areas, focusing particularly on mycotoxins related activities, adsorption, desorption for mycotoxin binders, degradation of mycotoxins, some surveys and biotechnological approaches towards mycotoxin remediation. I will briefly introduce some results as well. It's not simply a company pitch presentation, but I have some results to show as well. I kept the best part of the results for Mycokey to be organized in March. Hopefully we could see everybody in person and we can present some more work there. What we do as a company, our company is, we work in the area of animal nutrition. We provide animal feed and animal feed additives. We started our operations in 1991 from Republic of Serbia. And since then, we have grown to 200 plus employees. And with the help of 200 plus employees and 50 plus distributors, we are present on 6 continents, which is like more than maybe 35 countries currently. We also have our own labs in Republic of Serbia, and where we are performing mycotoxins analysis for animal feed materials, mycotoxin binder analysis, and other routine analysis for animal feed testing. What we do for international market is we provide innovative feed additives for in the area of mycotoxins and phytogenic based solutions. Some of our main product range are mycotoxin binders, particularly Minazal Plus and Minazal, and phytogenic feed additives are Ridomite, Petentaherba Plus, Discard, and Immunohype. Since it is mycotoxins related area, so I will mostly stick my talk on mycotoxins. So when I talked about these finding mycotoxins remediation based solutions, we are screening a lot of materials, either let's say, and trying different type of surface modifications for finding new binders or new materials that can bind mycotoxins. And we are doing it by performing adsorption, desorption studies. Adsorption is pH 3 and desorption at pH 6.5. And we have LC-MSMS from Agilent, and we are using that machine for routine testing, routine screening, and routine analysis of materials and developing some new binders based on that. We can also provide it as a testing service if somebody is interested for it. Other aspect we started in mycotoxins remediation is to find new microbes that can degrade mycotoxins. So we have been working on screening different type of soil samples from various natural locations to find microbes that can degrade at least the main mycotoxins, DON, giralenone, aflatoxin B1, ochratoxin, fumonisin B1, and T2. So all the analysis, again, I have been done by LC-MSMS, and whenever we were needing some help for metabolites analysis, we took the help of IFA Tulan and Dr. Michael Solyok there. So we use their laboratory services for finding some metabolites in our samples. So by screening various microbes, I don't want to present many slides here. We just found that some of the bacillus species which we found in our lab can, based on the metabolites which we detected, can transform or degrade 26% of aflatoxin B1, 25% of ochratoxin, and 95% of giralenone. So there is that type of, this type microbial degradation work which we are carrying on in our lab. We are still isolating more microbes, particularly related to DON and giralenone and other mycotoxins as well, just to see which one will suit to us and which one suit more in terms of applicability point of view. Since we have this LC-MSMS, I just want to emphasize one aspect of this food safety and feed safety, which is like testing everything but goes into food or feed. So we are trying to become part of that, and to do that, we have been screening just corn, which goes as a main component of animal feed, and we have been performing surveys. So like last year in 2019, we have found that 95% of samples were contaminated with one or more mycotoxins, and fumonisin B1 and B2, they were predominant mycotoxins, like 82% fumonisin B1 and 70% of the samples they had fumonisin B2, and in some of the maximums were also quite high. And just if I want to go back, we are also screening currently the corn which is harvested in 2020, and we are still getting some maybe quite similar results to last year, but we have not compiled it. So we hope to publish this survey for corn harvested in 2020 in early part of next year. We have our own microbiology lab where we are performing another aspect of mycotoxins based research is mycotoxin production technically. We all know that it's hard to find mycotoxins or enough mycotoxins for trials because of cost and other legal issues. So we are just growing some molds and we are trying to understand physiology of production of mycotoxins, and we want to use those materials for in vivo trials wherever possible. This is just an example of a corn contaminated with molds, which is just under incubation. So that's it. We are not doing this work alone. We are working with many fraternities of mycotoxins research community from various areas, from Spain, South Korea. Some are present in this conference, some are not, from Cranfield University, UK. So that's it from Petenco. It's a simple presentation. I just kept it simple, did not want to bore you much. So I just want to thank you very, very much for the listeners, to everyone, and please stay safe, keep your family safe, and hope to see you all in March 2021. Thank you very much, guys. Thank you. Hello.
Yes, thank you very much, Jan. There are no questions from the audience. So in your experience, which is the mycotoxin that you, as a target, could better offer performance in the degradation in your in this last years you have? What is the best work? Which mycotoxin was the best target for this degradation activity? In your last experience?
Yeah, we did lot of screening technically. We are still working on, let's say, on DON degradation and other mycotoxins as well. But we found activity only against aflatoxin, V1 ochratoxin, and girallanon. Currently, we are working on DON. We have found some promising microbes that can degrade DON. But again, you know, the safety and other aspect, further analysis, that all is in progress actually.
Okay. Thank you very much for your talk, and now we move to the next talk, which is Virginie Marquis from France. She's going to talk about efficacy assessment and protective effect of yeast fractions in reducing the negative impact of mycotoxins in animals, and Virginie works for Fileo Les Affres.
Thank you very much. Good afternoon, everybody. I hope you can hear me well and see my screen.
Yes, it's perfect.
Good. Thank you. So during this presentation, I will give you a short overview of Fileo, and I will present you some the benefits of yeast and yeast fraction in reducing the mycotoxin toxicity in animals, and I will present you some short results about the work done in the Mycokey project inside the work package 6. So at Fileo, we are committed to push the boundaries of animal care to better nourish and protect the planet. Our expertise, built over 150 years of research and manufacturing, is to bring the best of beneficial microorganisms to naturally improve animal nutrition. For example, we develop tailor-made solutions for preventive care, allowing a limited and responsible use of antibiotics. And for that, we improve and module the immunity and microbiota of animals. So we are developing solutions based on yeast, and today my talk will be focused on yeast. So first, a yeast is defined by a microbiologist by a single cell fungus, and yeast are known and exploited since 10,000 years now for brewing and baking. And the yeast are good to make bread, wine, and beer because they are good at harvesting energy from sugar without oxygen during the process named fermentation. And during this process, the yeast also can produce some flavors that are used for sensory and nutritional qualities. And also yeast are good to transform sugar in ethanol. But more than that, yeast have several roles in supporting health of people, animals, and plants. And as example, these roles consist of, one of these examples consists of antagonizing other microorganisms. So, for example, in animals, probiotic yeast can reduce the pathogen pressure, can stabilize the rumen, and modulate the microflora. And another role as feed additive for yeast is to be used as a source of protein, to be an immune stimulant, and the topics of my present today is its role in mycotoxin mitigation. And as you all know, mycotoxins have several toxic effects as immune toxicity, and they cause serious health issues. There are many examples of reduced performances, depressed growth, poor reproductive efficiency in animals, and also an increased susceptibility to pathogen and disease. So here the yeast is used as antimycotoxin and can have different effects. First, an effect is a preventive one by controlling the growth of fungi in the field, and also by inhibiting the mycotoxin production of these fungi. And secondly, in animals, yeast can help to remediate and to protect against mycotoxin. The remediation is achieved by either detoxification by adsorption, absorbing the mycotoxin and reducing the uptake of this mycotoxin in the body, but also by biotransformation of mycotoxin into less toxic metabolites, and also the yeast is able to protect the animal by improving gut health and immunity. And all starts with the selection of the right yeast strain and the right process of production that gives proven effects answering the needs. So I would like to share with you some examples. Here is a first study with on broilers chickens exposed or not to yeast cell wall. And we have looked at the intestinal morphology, at the jejunum morphology. So here you can see the in orange, sorry, the yeast cell wall can increase the villi-8 compared to the control in the jejunal mucosa. And also, if we look at the goblet cells and mucus thickness, we can see that the yeast cell wall increased strongly global cells and in the same way increase the mucus thickness. So we can conclude that the specially selected yeast cell wall is helping to protect the intestinal epithelium integrity. In a second example, I would like to show you a trial with broiler chickens exposed to aflatoxin B1 and ochratoxin A with or without yeast cell wall. And you can see that mycotoxin reduce the relative weight of lymphoid organs compared to the control in light green. And when you have the presence of the yeast cell wall in orange, you have an increase of this relative weight. Similarly, if we looked at the immune cellular response. And first, regarding to the cutaneous delayed hypersensitivity, you can see that mycotoxin reduce in the dark green the delayed hypersensitivity, and with the yeast cell wall, you have an increase. Similarly, mycotoxin reduce the concentration of lymphocytes, leukocytes, and heterophiles in the blood. And in presence of a yeast cell wall, you have an increase, and it counteracts the effect of the mycotoxin for the content of lymphocytes and leukocytes. So in conclusion, mycotoxin reduces the immune response of broilers, and the addition of the selected yeast cell wall can be an alternative to counteract the negative effect of this mycotoxin in broilers. I would like also to present you the yeast cell wall fraction in adsorbing agent. So mycotoxin can decrease by the mycotoxin bioavailability can be decreased by adsorbing agent and by the yeast cell wall. In this yeast cell wall, you have the presence of a network of beta-glucans, chitin, and manoproteins. And this three-dimensional structure of these components help to adsorb mycotoxin by intermolecular interactions. And important characteristics of this yeast cell wall are polarity, hydrophobicity, size of accessible surface, and the flexibility of this yeast cell wall to better adsorb the mycotoxin. There are several methodologies to evaluate this adsorption, and we decided to measure it in vitro first to screen a lot of yeast cell wall and select the best one. And we do that by incubating the yeast cell wall in different buffer, in different conditions, and realizing an isotherm to evaluate the adsorption parameters of the yeast cell wall. Then we confirm these results in vivo by doing in vivo toxicokinetics. And finally, we confirm the efficacy of our product in vivo by doing in-field animal trials. So I would like to share with you just a short example of what we do in this Mycokey project. We have worked in the work package 6 with Ispa and Ghent University to develop a new yeast cell wall. The objective was to improve the capacity of the yeast cell wall to increase the adsorption of the mycotoxin. So here I present only the results with zearalenone. And you can see here that the adsorption of zearalenone is above 80%, whatever the pH. We have then realized the adsorption isotherm and do some desorption study and confirm that yeast cell wall has high efficacy in adsorbing zearalenone at acidic pH, and that the yeast cell wall retain the mycotoxin when the pH rises to neutral values in intestine. Then we have realized in vivo toxicokinetic study with Ghent University, and we have it's a trial in piglets where animals were exposed to a single oral bolus of zearalenone in 2 conditions of protocol, after feed withdrawal or when feed was added ad libitum. And we can observe that there's a reduction of the area under the curve of the metabolite then glucuronide. And finally, we observe that there's a reduction of the bioavailability of the mycotoxin. So we have confirmed that the yeast cell wall product was able to reduce the systemic exposure of animals to the mycotoxins. So in conclusion, yeast and yeast derivatives can act at different levels to manage mycotoxin risk at prevention, remediation, and protection levels. it is also crucial to select the right yeast or yeast derivative based on expected outcome. And finally, yeast is a sustainable solution to manage health through nutrition. So I would like to acknowledge the partners in the Mycokey project, and in particular the Ghent University and Ispa for their help in this project. And thank you for your kind attention.
Thank you very much, Virginie. Very nice presentation. There are no questions from the audience, but I have a curiosity. This ICV seems to work very well on the animal, toward the animals. Do you think we could ever think about the possibility to use it for humans?
Yes, I think it could be possible, but here it's maybe a problem of regulation. But yes, yeast and yeast cell will have a good probiotics effects in humans. So why not use it also for mycotoxins?
Okay, thank you very much. Thank you. And,
thank you very much.
I move to the next speaker, that is Marie-Clarisse Techer of Mixed Science from France. She will talk on biological detoxification of zearalenone by Bacillus subtilis strains.
So I think, yes, thank you very much. I think you can see me, and I had to share screen, that's right?
Yes, you should share the screen. Okay, we can see.
Okay, very good.
Thank you.
Good afternoon, everyone. I'm Clarisse Techer, a research and development manager in Mixed Science in Olivet, and I'm pleased today to present some results obtained in a collaborative work with Ispa, so the Institute of Science of Food Production in Bari, Italy. My talk will focus on biological detoxification of zearalenone by Bacillus subtilis strain. So first, a few words about the context of the study. As we know, mycotoxins are produced by certain mold in specific environmental conditions. It is generally small molecules, and some of them are stable at high temperature. Mycotoxins show wide chemical and toxicological diversity. Major mycotoxins of interest in feed are aflatoxin B1, deoxynivalenol, zearalenone, ochratoxin A, and fumonisin B. Sorry. Oops. They may have significant economic impact with the loss of feed in the industry, and there are different consequences, as we have seen before, in the food and feed at different levels of the value chain. In livestock, for example, they can reduce performance of animals, induce reproductive issues, and in case of acute toxicity, lead to mortality of animals. A major point of mycotoxin effect is the sanitary issue in humans, especially for some mycotoxins such as aflatoxin B1, which is recognized as carcinogenic compound. So mycotoxin decontamination can be worked at different levels using physical or chemical methods and by the use of different agents in the feed solution. For this last strategy, two major types of compound can be used. Compound with indirect activities toward mycotoxin, such as antioxidant agent, immunostimulatory agent, which may support global animal health. And the second type of compound, the most commonly used, are those which may have a direct activities toward mycotoxin, allowing also to decrease their bioavailability. Main examples are adsorbing agent. Less used but promising are biotransforming agents. These compounds involve mechanism of mycotoxin degradation into non-toxic metabolite by enzyme and or microorganisms. And these common activities are specific to some microorganisms and have been described by fungi, yeast, or enzyme. among potential biotransforming agents, we have E. coli bacteria. So the objective of the present study was to investigate the efficacy of the 3 Bacillus subtilis strain in reducing mycotoxin, especially the adenine and its major metabolite of phase 1 in culture media. The experiments were performed to answer to different questions. Have the Bacillus strain a potential for mycotoxin detoxification? And if yes, what are the optimal conditions and what could be the mechanism involved in mycotoxin detoxification? First, some experiments were conducted in order to evaluate the potential of the 3 Bacillus strains of interest towards the main mycotoxin, such as zearalenone, aflatoxin B1, fimosine B1, deoxynivalenol, ochratoxin A, T2, and HT2. Results are presented in this table, and it shows that percentage of reduction of zearalenone were high for the 3 strains, varied from 85% to 95% according to the strain. So these first results were promising and show that the 3 strains have a good potential for ZR reduction. Further experiments were performed to check if the strains were effective against ZR metabolite. Indeed, it's important to check because some metabolites are more toxic than parent molecules. The results are presented in this table, and we can observe that the 3 Bacillus subtilis strains were effective for ZR major metabolite reduction. Reduction percentage varied from 80% to 100%, according to the tested strain and the metabolite. Concerning the condition for ZR degradation, on this figure, we can see that the percentage of ZR reduction in function of the time for the 3 Bacillus strains. So what we can observe and what is important in this figure is that the plateau is obtained between 10 and 16 hours, suggesting also that ZR reduction by this bacteria is fast. The next figure shows the ZR reduction according to different conditions of temperature and pH. And briefly, we can observe that the Bacillus strains were effective in a large range of temperature from 20 to 40 degrees and pH 5 to 8. Concerning the mycotoxin concentration and medium composition, we have shown that our strains were effective in a large range of their concentration and in different media, including those containing the toxin as sole carbon source. And after some trial optimization for bacterial growth, the efficacy of the strain were evaluated in anaerobic conditions. So it's quite an important criteria because it's a main condition, for example, found in fermentation process or in digestive tract of animals. So results show that the ZR reduction were higher than 80% in media in anaerobic condition. Then, to understand the mechanism for ZR detoxification by our Bacillus subtilis strain, ZR removal by culture, cell pellets, and supernatant were evaluated. As presented in this figure, bacterial culture and viable cells show significantly higher ZR removal efficiency than supernatant, suggesting also that these activities were intracellular and are to release into the growth medium. And to finish, to understand the mechanism of ZR detoxification, some adsorption and desorption trials have been performed. As we can see with black bars with ZR desorption figure, desorption extracted little amount of toxin, maybe about 10% from the cell pellets, in comparison with ZR reduction with viable cells on the figure above. This result indicate that viable cells of Bacillus subtilis strain are able to metabolize very quickly most toxin in the buffer. And interestingly, both acid and heat-treated cell reduce similar amount of toxin from the buffer. And desorption trial using these cell pellets from treated cells recovered most of toxin removed by these not viable cells, as we can observe from the figure on the bottom. So this experiment shows that the removal of toxin by viable and not viable cells of Bacillus subtilis strain occurs at the same extent, but by different mechanisms. A metabolism of ZIA occurs with viable cells, while binding of the toxin to bacterial surface is the mechanism underlying the removal of toxin by dead cells. So in summary, this study has shown that the three strains of Bacillus subtilis were efficient for their detoxification and their measured metabolite in various conditions, and that their removal was fast in less than 24 hours. Concerning the mechanism, we have shown that enzymatic activities which may involve in this reduction, and that the desorption extraction trial of the mycotoxin from pellet suggests that their removal by Bacillus subtilis strain involve different mechanisms according to the strain and the state of the cell, so live or dead. So all of these characteristics suggest that Bacillus subtilis strain could be exploited in food and feed chain to reduce mycotoxin contamination. So many perspectives of work can be explored in this project. First, in a scientific point of view, literature lacks of information on the metabolite produced by Bacillus subtilis strain during the removal of the toxin. Further studies are also needed to identify this metabolite and to assess the residual toxicity. Also, research perspectives could be the identification of gene or enzyme involved in metabolization. And secondly, in an applied research point of view, it could be interesting, and it's in progress, to investigate these activities with in vivo trial. And as we know, mycotoxin symptoms are many and diverse, resulting from a cascade of events. So it could be interesting to explore other key functionalities which may help animals to counteract negative effects of mycotoxin, such as antioxidant or immunostimulant agent effect. So thanks again to Ispa for this work, and to my colleague from MixScience and Nolivad. And last but not least, in MixScience we say safety first. So safety is health. So take care of yourself, and see you next year for the second part of the Mycokey events. It will be a pleasure for the MixScience and Nolivad teams to meet you and to present our product portfolio on this important topic. So thank you for your attention.
Thank you very much, Marie-Clarice. There is a question from the audience.
Yeah, there are two questions. One from Bert from Bremen. He asked, how can this be applied in practice in animal feed?
There is many application which can be possible with this bacteria. We can think about, for example, a formulation or the use of this compound as additive, so in the feed solution. Yes, maybe the main application could be this one.
There is another question from Michelangelo Pascale. Bacillus subtilis, is it degradation or biotransformation? Did you start with the characterization of products after incubation with bacterium?
So we think that maybe it's degradation, or we are not really advanced in this topic, but we have the hypothesis that it could be lacase, some enzyme produced by Bacillus, just a lacase, which can involve and induce this metabolization. But we have to check, and we have to go deeper in experiment.
Thank you.
Thank you very much, Marie-Clarice, for your presentation. And now we move to the next speaker, Gerardo Morantes from Bühler, Switzerland, and he will talk about advanced grain cleaning solutions for mycotoxin reduction. And Gerardo, I hope I pronounced well the name. I don't know if it's Gerardo or Gerardo.
It is fine.
Okay.
Gerardo is fine.
Okay.
Thank you very much. Thank you to the Mycokey conference for the opportunity to talk about our company and talk about some of the results of the collaboration that we have been taking with Ispa. And so my presentation is about mycotoxin reduction by advanced grain cleaning. And my name is Gerardo Morantes, and I am Director of Food Safety for Bühler in North America. And so I am speaking from Minneapolis this morning. It's snow in Minneapolis already. And I would like to speak a little bit to begin with about Bühler and what Bühler is. Maybe you're not familiar with it, but every day billions of people come into contact with Bühler. And we are, Bühler is a technology and solution provider company producing machines for food and feed production, mobility, and communication. And during the day, you will probably come in contact with products that have been produced on our machines at least once, either by the breakfast cereals in the morning produced from our extruders, or the coffee produced from beans that have been roasted on our machines, or maybe the beer in the evening produced with malt that went through our malting solutions. So Bühler covers a whole range of areas of the food supply chain, and also in the mobility and advanced materials space. At a glance, Bühler is, is a company that is 100% family-owned company, 160 years of existence, and with more than 12,000 employees spread out around the world in 140 countries, and with 32 manufacturing sites around the world and service stations. One important fact to highlight is that up to 5% of the turnover the company is invested in research and development. And we have some sustainability targets that are very important. Because our question, is how to sustainably nourish 10 billion people by 2050. We are very aware that we are using today 1.7 times the resources of our planet Earth, and that creates a huge problem. And part of the problem is the losses that are created throughout the food and feed supply chain. And some of these losses are due to mycotoxin contamination. Therefore, we have set very specific targets to reduce by 50% by 2025 energy, waste, and water in the solutions that we are providing to our customers. So we do this in partnership with our customers, obviously, and with our employees. And waste, which is a big part of our objectives, is very much connected to the mycotoxin problem. In general, we can see that our approach to mycotoxin control is to the whole value chain. We don't see the mycotoxin problem being solved by one specific action, but a combination of actions throughout the whole value chain that starts from the farming and the harvest with good agricultural practices and goes through the different steps that the grains go when they are being utilized in food processing lines and feed processing facilities. And at the end of the day, our best bet is in being able to prevent that the contaminated materials make it to the consumer side. And so we try to reduce that concentration along the value chain. Secondly, the use of the right cleaning technologies is very important, because only a small fraction of the grains carries most of the mycotoxins. Thanks to the research and experience, we know that these fractions are namely the dust and the broken kernels and the lighter fractions of the grains. And that knowledge helps us to remove these highly contaminated fractions through the combination of mechanical and density cleaning combined with aspiration and optical sorting. In the work that we have done with Ispa, we have tried to combine the use of efficient cleaning technologies to remove the contaminated fractions. And how can they be removed? Well, typically the dust and broken kernels contain very high contamination levels, which can be removed by sieving machines. We combine inlet and outlet aspiration. And here we can also remove coarser foreign materials. Mold contaminated kernels are often lighter than the healthy ones and can be removed by density separation in the concentrator. This makes sense because, generally molds will grow in kernels which have been attacked by insects. And so if the insects are consuming some of the nutrients or the fractions of the kernel grains, then they will be for sure like kernels. And finally, grains with visual fungal features such as color defects can be removed by optical sorting machines. In our case, they are called Sortex machines. In Sortex, in a nutshell, can do what we cannot do during sampling. The machine looks at every single kernel, and is like a policeman who makes sure that all kernels which carry signs of contaminations are removed from the stream. Also, a new technology based on fluorescence is just being developed, focused on the removal of aflatoxins in maize. And this is a technology that we call LumoVision, and it uses the spectral data of fluorescence to sort out aflatoxin contaminated kernels in maize. And it is, like I said, in development, it's still in development, it's still in pilot testing, both in Africa and India, where there is a great need for this type of solutions. And we will continue to present updates on the development of this machine. But basically, the machine, we have found a high correlation between the spectral data with the fluorescence of the color of the kernels and the contamination levels. And by using a highly sophisticated fluorescence camera, we can detect this fluorescence in the kernels. And at the same time, this is revolutionizing the area of removal of aflatoxin contaminated kernels, because it also is automated and uses digital information to really being able to develop these correlations and being able to, in practice, being able to remove a significant amount of contaminated kernels, and in that way preventing the aflatoxin contaminated kernels from getting to the consumers. As a take-home message, I would like to say that we recommend for a successful post-harvest mycotoxin reduction, first of all, being able to obtain representative samples. Sampling, as you all know, as everybody involved in this topic knows, is one of the main challenges to be able to detect contaminated streams of grains. Then after that is to be able to do the proper drying and stabilization of the product and have safe storage solutions to avoid contamination. Removing then all contaminated fractions by grain cleaning, removing the coarse, the fine, and the low density impurity, and then using optical sorting of discolored or defective grain kernels to complete the process. In the end, expertise and knowledge is really key in this complex topic. But of course, also, we cannot do this alone. And that's why we have been partnering with science, the scientific community for effective mycotoxin risk mitigation. And we have partnered with Ispa for industrial scale grain cleaning studies. And thank very much the team at Ispa for the collaboration and the support. And we are also partnering with other institutions to continue our research, and we will hopefully be able to present some of these results in the coming year. And finally, I want to thank you for your attention, and want to provide my email address in case anybody needs any additional answers after this conference, or any additional information on the topic that I have spoken about. Thank you very much.
Thank you very much, Gerardo. And there is a question from the audience.
Yeah, there is a question from Joanna. Can fluorescence be used for sorting other grains as coffee?
In coffee, it might be done in coffee, but at this point, our efforts, you know, when we are doing R&D, we have limited resources, right? And we have focused the effort on the corn, given that the corn is one of the largest commodities in the food and feed supply chains, and the impact of the aflatoxin in the human consumption is so big in animal consumption. Therefore, our initial focus is on maize at this point.
Thank you very much, Gerardo. So thank you all speakers of this session, the Voices from Food and Feed Industries. And now it was my big pleasure to introduce you Rudi Kurska from Austria. He's the leader of the other My Toolbox, my European project on mycotoxins that has been funded by the Commission in the last call. He will talk about the impact of integrated management strategies as proposed by My Toolbox to control mycotoxin in the food supply chain. Rudi is also the president of the International Society of Mycotoxicology. We will have at the end of this conference a meeting of ISM always using this platform, and is our friend since more than 30 years. So it's a great pleasure, Rudi, to give you the words for your presentation.
Yeah, many thanks, Antonio. I trust that everybody can hear me.
Yes.
So if not,
yes.
Yeah, thank you. Yeah, I don't know whether you can understand me, but at least when you can hear me, that's already 50% of what I would like to achieve today. Thanks, Mr. Vice President of the International Society of Mycotoxicology, which is already kind of an announcement of what is to come. Namely, also we have an ISM meeting later on. But now I'm very glad to give a presentation on the impact of integrated management strategies as proposed by my toolbox to control mycotoxins in the food supply chain. As you know, there have been two major projects which have been funded over the last 4 years by the European Commission. My toolbox on the one hand and Mycokey on the other hand. I'm very thankful also to my friend and colleague Antonio Logreco and this whole team that they have also involved my toolbox in these Mycokey events during these days. We have here kind of evidence why mycotoxins do matter from a food safety standpoint. There are numerous reasons, as mentioned before during the course of the conference, including certainly all sorts of health effects, agricultural losses, which go in billions of euro or dollars around the world. But this is one of my favorite pictures, which shows actually the rapid alert system for food and feed. So the early warning system established in Europe, which actually clearly indicates that it's neither heavy metals nor is it pesticides, which are really highly recognized much more than mycotoxins, nor pathogenic microorganisms. It is actually mycotoxins, these fungal secondary metabolites, which are toxic, which account for the greatest number of rejections due to this contamination at the EU border. And that has most likely been one of the motivations the European Commission had to fund projects or to launch a call asking for tools along the whole food and feed chain. And in the My Toolbox approach, we have been pursuing an approach which starts at the left lower corner of this slide here, with pre-harvest techniques, clearly including biocontrol, resistance breeding, also early warning systems, post-harvest techniques, including the decision support systems for storage of kernels, food processing to start to check and to study the fate of mycotoxins during baking, and then also the safe use options of contaminated batches in order to be able to make the most out of contaminated batches, such as the production of biofuels and dried distillers grain soluble. So that being said, I'm very thankful to all the work package leaders. You see very similar, very familiar faces with Simon Edwards, Ferenc Bagi, John Gilbert, Carsten Hasak, Michaela Pichler, Una McMurray, Monique Denise, Birgit Boschmeier, who was my project manager, but certainly also old friends like Narish Megan or Michele Suman from Barilla, and last but not least also Vittoria Rossi and Ine van der Vels-Klerk, who took care of these structure which I have depicted in my previous slide, in which was then executed through a number of work packages. So you see, the kickoff meeting dates already back to 2016. Project finished actually in March this year. So we had a consortium composed of a total of 23 partners from 11 countries, and similar to Mycokey, we also had a Chinese participation with 3 strong partners, and I will allude to that or come to that aspect in depth later. Starting with pre-harvest. This is a slide Ferenc Bagi kindly provided, and which is a slide testifying to the fact that we have been dealing with the use of atoxygenic isolates of Aspergillus flavus in Serbia. Serbia is the 10th largest maize exporter in the world. You can also see in this pie diagram that they have a pretty much focus on primary products of some 80%, which is quite impressive, and of processed products of about 21%. So that's together with this so-called maize scandal dating back to 2013 as a result of heavy aflatoxin contaminations in maize, has been another motivation, not only I think for the European Commission to launch that call again, but also for us to focus on Serbia and to particularly screen, select, and characterize atoxygenic isolates of Aspergillus flavors, which could be used as biocontrol agents in Serbia. And these, how they called it, aflatool was applied actually when maize had 10 fully developed leaves, 10 kilogram per hectare. You see here these sorghum seeds, which were inoculated with the atoxygenic strain of Aspergillus with the aim to outcompete the toxic strains of Aspergillus on the field. And that was done in various areas in Serbia. You see in red the non-treated maize samples, in yellow the treated ones, and certainly we also had the buffer zone in between, which is indicated in gray color, both in irrigated and in unirrigated conditions. Certainly the unirrigated conditions being the greatest indicator for the success of these biocontrol agents, since unirrigated conditions resemble the best the drought conditions which caused actually the aflatoxin issue some years ago. So you see on the left diagram that in the year 2017 indicated with red bars, and 2016 indicated by the green bars, that in all cases where it's irrigated or unirrigated, the treated samples have significantly reduced levels of aflatoxins, and we had in 2018 in these regions there was hardly any contamination with aflatoxin B1, so that's why they moved to another area. And here you can see that in 2018 in Uljama, the untreated sample, or the treated sample vis-a-vis the untreated sample, was again very successful as a result of the application of these atoxygenic strains. So that being said, I would like to switch gears, come to another aspect of our project, which is in fact the fig contamination. So we are stick with aflatoxins for now. One of the issues is aflatoxins in figs imported from Turkey. Despite a lot of quality control, there's room for improvement, especially getting away from the practice that these contaminated figs are selected from the non-contaminated figs via UV light under very harsh working conditions. You can also see, as you have seen on the previous slide, that on the figs also irradiate some fluorescence as a result of aflatoxins being present there. So we have actually, that was done in a work in Graz in southern Austria, EVK, together with Insort. There was a sorting machine coupled actually to a hyperspectral imaging device, and you can see actually on these pictures the red spots indicate the presence of these fluorescence molecules that contaminated figs could quite nicely be separated from non-contaminated, the blue spots vis-a-vis the orange spots with an accuracy of about 80%. So that was also a very nice result. Moving on to storage, we have had a very successful collaboration with ASAG from China near Beijing. It is just unbelievable, and that was also kind of very fascinating from my angle, from my point of view as a coordinator, and for all the other participants, that each of these buildings contain 8,000 tons of wheat or maize, respectively, which are stored in case of wheat for up to 5 or 6 years. And that is why in China, because they want to brace themselves for any crisis which might come up, that they are interested in improved monitoring and decision support tools. And that's why we have had the chance to also go inside these storage facilities. We are actually on the top of some 8,000 tons of wheat, walking along, and you can see on the right upper picture that these are the sensor nodes to measure carbon dioxide, temperature, and humidity, and then to link these data to biological data and fungal growth. Very successful experiments have been carried out at Barilla site with wheat, with these sensor nodes clearly indicating that this is a very good approach to come up with something like this, that you can then look into the storage and see in red, due actually to CO2 being a much better earlier indicator for the presence of fungi and mycotoxins, that if there is a rise, you can have a red or yellow alert, and then decide whether you spray pesticides or do aeration or remove the kernels. In China, we have only had the chance to look into peanuts due to some technical and organizational issues, but nonetheless, this collaboration was very successful. Here's another very nice. I'm sorry for some messed up slides, obviously, to this connection and formatting whatsoever. Not all slides are exactly the way presented as they are supposed to be, but I think it should be good enough for you to get the gist out of it. We have also been working on innovative milling strategies with Barilla, which actually has led to a 3% higher fiber content. Actually, so due to innovative approaches in hammer milling systems and combination with sieving shifters, we have been able to increase the fiber content significantly. You can see actually here that the whole grain pasta, the commercial pasta, could significantly be upgraded to whole grain pasta by still containing very low levels of deoxynivalenol, which is usually a potential issue for whole grain products. So that being said, sticking with Barilla, we had a very good collaboration, actually a very exciting collaboration, in order to study the fate of mycotoxin during industrial baking processes. So we looked into bread, we looked into crackers and biscuits, and spiked these food commodities both with C12 isotopologues of deoxynivalenol as well as C13 isotopologues. And by doing that, we are able to extract only those degradation products which are real degradation products and not only artifacts as a result of high resolution mass spectrometry, which produces unfortunately some 95% garbage signals. So by doing so, by utilizing these so-called tracer fate studies with the C12, C13 approach, we were able to look into corresponding pairs of potential degradation products, and then to do a full mass balance, which showed us that actually the reduction of deoxynivalenol was achievable by a level of 2 to 6%, which is not as high as some of the existing literature might testify to, but all the degradation products, like especially isodone, are non-toxic. One of the aspects which I also wanted to bring your attention, and again, sorry, this slide is messed up for whatever reason. You see the black line, which shows the calibration curve when you do the analysis, and then with standards, and then you see in green the one which is the one which you obtain by observing a signal separation of 50%. The dashed red line is supposed to indicate that there is, in addition to the well-known issue of signal separation effect in mass spectrometry, also a relative matrix effect as a result of lot-to-lot variation. And that is actually a nice picture showing you that both for maize and figs, you see on the x-axis single lot and lot-to-lot, that actually within a lot variation is much lower than lot-to-lot, which means that this is an underestimated issue in mycotoxin analysis, that indeed the lot-to-lot variations have to be also considered for mycotoxins when expressing the result with the uncertainty. And that is, to make a long story short, when you calculate the expanded uncertainty, we actually came up with a 50% proposed uncertainty for mycotoxin analysis, which we think should be applied in the future, and which is actually also quite in the area which Thomson has published in the year 2000, but based on ring trials. That is all has all been published in Food Chemistry. And finally, I would like to spend a few words on safe use options and detoxification. We have our company partner Biomime on board. We have these recombinant enzymes which can convert formonicin into hydrolysed formonicin B1 and serralenone into hydrolyzed serralenone, which also EU authorized as feed additives, and which were actually kindly provided in the bioethanol area, where it is the bioethanol, the biofuels, which is produced, and where, however, the whole process is only economically sensible or feasible when the byproducts, like the dried distillers grain soluble DDGS, which can be used as a protein-rich fraction in feed in the feed area, can also be produced free of mycotoxins. And that was achieved by adding actually these enzymes, mycozymes, prior to the saccharification and also to the fermentation. These were different trials showing that formonicin was reduced by 99% and serralenone by 89%. The biotransformation was also confirmed by the formation of these degradation products, which I have mentioned to you. So finally, we have also been dealing with feeding trials. This is something where such feed additives, which were also applied in this bioethanol production system, were also applied during feeding trials. But there is no common scientific basis between EU and China on the efficacy and which kind of biomarkers to be used to test the efficacy of these feed additives. On the one hand, to study aflatoxin B1 absorption in dairy cows, and on the other hand, formonicin B1 reduction in pigs. And actually, although we had some sort of issues around getting the detailed report from China, we can really conclude in a very positive way that what we have done is unprecedented. We have indeed carried out for the first time in vivo experiments to test the efficacy of feed additives under local conditions in China, never been done before. according actually to this European Commission regulation for evaluation of feed additives. So this trial indeed, we think, marks an important step towards establishing a common scientific and also ethical basis for feeding trials in the future, and this also will save costs in the future between EU and China trade. And well, we have also been there. It shows also Veronika Nagel and Gerd Schatzmeier and myself. We've been to the facilities for swine and cattle, and had a very good collaboration with our partners from the Feed Research Institute of the Chinese Academy of Agricultural Sciences. Last but not least, or actually last, but best, the e-platform, similar to Mycokey approach, but we had here the idea of splitting all the information and also tools developed into an e-platform consisting of a static part providing information and a dynamic part providing actually sensor data that was coordinated by Wageningen, by also actually including documents which are usually in a not very friendly format, in a user-friendly format in this toolbox, so that actually people can look into cultivars, what is the best way to for tillage and crop debris utilization, which kind of biopesticides are there, etc., doing some scenario analysis, and also then in the dynamic tool to look into forecasting models, so that actually based on previous crops used on this field, soil tillage, weather station data, and also with my GPS data, I get a yellow, red, or alert or green information that in my area I'm safe or there's a risk for mycotoxin contamination. I'm very glad that a company Biomine, now DSM, has licensed this toolbox and the forecasting system, so that this is an activity which will be going on and utilized also beyond the lifetime of this project. Finally, to summarize, we have actually achieved for the adoxygenic strain some 90% aflatoxin reduction in the Serbian maize fields. For rejections of dried fields, 80% rejection accuracy was achieved using one camera only. That is an aspect which can be improved with additional cameras beyond this project. A decision support system for pilot silos for peanuts in China has been successfully installed. Innovative milling strategies were developed and applied, like whole grain pasta with 3% higher fiber content and improved fiber-dawn ratios. The fate of dawn during baking under industrial conditions was successfully studied, and a full mass balance is for the first time available for thermal processing of mycotoxin contaminated grains in production of bread and crackers. We've also successfully applied recombinant enzymes to reduce bioethanol by degrading these mycotoxins and coming up with toxin-free DDGS. And the EU-China collaboration led to the efficacy of feed additives tested under local conditions according to EC regulations, and also the E2 platform was developed. With that, I would like to thank my team for the enthusiasm and the team spirit, and would also like to say that as a coordinator, it was fantastic. I'm glad I did it, partly because it was worth it, but mostly because I shall never do it again. Well, this is certainly a joke. It has been a great pleasure to work with all these guys, and also to do some farming myself with minimum, and also with all sorts of blowing techniques, which was a great experience. So thank you very much for listening, and thanks also to you, Antonio, for being a great chair.
Thank you very much, Rudy, for your usual brilliant, very brilliant presentation, and excellent result you have shown that were achieved in my toolbox. There are two questions from the audience, so Alessandro, please.
Yeah, there is a question from Adil Muana. Does the UV screening to evaluate mycotoxins in figs requires any pre-treatment for the sample?
No, this is that's the speciality. So this is something where you just have a sorting machine, and then these figs are traveling along a moving belt, then fall down, and once they fall down, they are monitored by a hyperspectral imaging camera, and if the hyperspectral imaging camera detects any contamination as a result of fluorescence, then a flip will open and they will remove these contaminated figs. Having said that, it has to be pointed out that this is not a direct determination of the aflatoxins, because there is also the shape. There is also some other fluorescence compounds like kojic acid. So there is some limitations to that. However, based on the requirements of the customers of InSort, what they told us, it is not the aim of the customer to know exactly the amount of the aflatoxin in the fig, but to be sure to eliminate anything which is potentially contaminated. So, but yes, no sample preparation, this non-destroyable method.
Okay.
And there is the last question.
There is another question from Susa Vogelsgang. Okay. For which mycotoxins and crops could the DSSE platform be used?
The decision support system is actually now available for DON, for zearalenone, for ochratoxin A, and for aflatoxin B1.
Thank you very much. Okay, so thank you very much again, Rudy, and see you later for the ISM.
Talk to you later at the
assembly.
ISM.
And I'll give you now the words to Alessio Bacchelli from the European Commission, and he will launch us in the perspective of the Green Deal call from FARC to fork. Alessio.
Thank you, Antonio. I don't know how, show my screen. That's it, perfect. So thanks to all for this final conference, for inviting me. I hope you can see my screen.
Yes, we can.
Okay. So I will make a presentation on the Farm to Fork call within the Green Deal call package that was just launched. I will cover first the policy aspects of the Farm to Fork area, namely the topic 6.1, and then speak about the implementing aspects of the call. So we can start. So basically, the Farm to Fork strategy is paramount as a component of the European Green Deal, and addresses comprehensively all the challenges of sustainable food system by recognizing the inextricable links between healthy people, healthy societies, and a healthy planet. The Farm to Fork strategies also tackle some of the sustainable development goals. And of course, is also part of the just transition mechanism that is another key aspects of the European Green Deal. So as you can see, the Farm to Fork strategy tackles the three dimension. So sustainable food systems ensures at the same time environmental, social, and economic sustainability, and also has specific goals, 4 goals. One about the climate footprint, so to reduce the environmental and the climate footprint of the food system. Lead into a global transitions, so to have a competitive and sustainable farm to fork food systems. Also to tackle on what could be new opportunities, and of course to create a more resilient food system. The farm to fork strategy has also some targets that should be achieved by 2030. This can be found with more detailed explanations in the Green Deal website. So one of them is about to reduce by 50% the overall use and risk of chemical pesticides, and of course, reduce by 50% the hazardous pesticides. Reduce the nutrient losses to at least 50%, but at the same time ensure that the soil remain fertile or increase its fertility in a way that there is a reduction of fertilizers to be used by at least 20%. And also reduce the sales of antimicrobials for farm animals and also in aquaculture by 50%, and make sure to use at least of the 25% of EU agricultural land with organic farming, and of course use more and more organic aquaculture. Now, this is the topic you can see in the funding and tender portal. It has this code, the horrible code, but it means LC stands for low carbon, GD Green Deal. 61 is simply the numbering of the topic, and the title is testing and demonstrating systemic innovation in support to the farm to fork strategy. It's an innovation action. And there are 74 millions of euros allocated to fund many projects. We don't know how many, but for sure 6, if not more. And you can see, if you read the Green Deal call introduction section, it says that it should deliver tangible and visible results relatively quickly and show how research and innovation can provide concrete solutions for the Green Deal main priorities. So what we want is to fund projects that could deploy results quite quickly. This is why it's also an innovation action. This topic addresses the three component, the three perspective of sustainability: social, environmental, and economic. You can see, for example, it tackled climate change, it tackled the just transitions, it speaks about social rights of workers in the food chains, and so on and so forth. And then we go in the scope. The scope is the main part of the call. It addresses the global challenge. The idea is to support deployment and scaling up of innovation that contribute to the objective to the farm to fork strategies. Activities and outcomes actually should tackle TRL between 5 and 7. You can find the description of the TRL again in the Horizon 2020 general annexes of the work program. And it tackles these 6 subtopics. Each subtopic actually is a food system. Some of them are more popular than others. So the first one is A, achieving climate neutral farms by reducing greenhouse gases emissions and by increasing farm-based carbon sequestration and storage. The B is achieving climate neutral food businesses by mitigating climate change, reducing energy use, and increasing energy efficiency in processing, distribution, conservation, and preparation of food. Reducing the dependence on hazardous pesticides, reducing the losses of nutrients from fertilizers towards zero pollution of water, soil, and air, and ultimately fertilizer use. Reducing the dependence on the use of antimicrobials in animal production and in aquaculture. Reducing food losses and waste at every stage of the food chain, including consumption, while also avoiding unsustainable packaging, and shifting to sustainable healthy diets sourced from land, inland water, and sea, and accessible to all EU citizens, including the most deprived and vulnerable groups. The topic requires to choose one of these 6 subtopics, one of these 6 food systems, that you can see they overall encompass all the main challenge, all the scope of the farm to fork strategy and the 2030 targets that I spoke before. There is an indication about the proposal size. We aim at funding proposals between 6 and 12 million to be reached under these subtopics. And also we will have a common ranking list, and we will have to fund, the European Commission will fund the highest ranked proposals per subtopic. For sure, each subtopic, each food system will have a proposal funded as long as this proposal has attained all the provided thresholds. Now, again, a bit on the scope. The proposal should focus on the systemic innovation and also should pay attention to particular aspects. I'm not reading them all, but you can see they require to be very holistic, to use the multi-actor approach, to make use of SMEs, the young professionals, consumers and citizens are also important to have this also inputs, feedbacks from that particular stakeholders, and also not focusing too much on research activities, but rather on addressing the gaps for solution building, testing, and demonstrations. And of course, capitalize on existing demonstration facilities. Expected impacts. Again, as each Horizon 2020 topic, there is a structure, and after the scope, the challenge, there is the list of the expected impacts, which are very briefly these three, to demonstrate that these innovative systemic solutions have the potential to generate significant positive impacts by 2030. Remember the 4 targets of 2030, to contribute significantly to the achievement of the objectives and targets of the Farm to Fork strategy for the European Green Deal, how achieving these targets within the Farm to Fork strategy can be a vital piece of the puzzle for the European Green Deal. And also, of course, increase the the awareness of policymakers, businessmen, investors, entrepreneurs, institutions, stakeholders in general, including the citizens, in promoting and realizing the uptake at youth scale for the behavioral change of the Green Deal. Now a bit of aspects related to the implementation. So the call deadline is next year, 26th of January. We have as a target, as a standard in our agency, but set by the Horizon 2020 regulations, 5 months of time to give the results of the evaluation, so to inform the applicant. And then we have some other 3 months to prepare the grant agreements. So basically, the projects will kickstart not earlier than end of September, beginning of October, the selected projects. This is in common with the other Green Deal topics you might be interested in applying as well. So how the evaluation works, by the 26th of January, we receive the proposals, we do the admissibility, eligibility check, we allocate the proposals to evaluators, we make a briefing to the evaluators to explain how the evaluation works. They carry out in their own capacity the individual evaluation, then they meet. They will not meet physically, from my understanding, if things continue as they are for the COVID, but we will organize virtual meeting like we are doing now with the webcams and with this web conferencing tool to reach a consensus on the report. There will be a panel review with the list of the ranking that will be discussed. And then we will launch this ethics screening review. And then we will send out this evaluation summary report and invite at the same time those in the main list to prepare the grant agreement. Now, this is only an innovation action. Innovation action are funded 100% to every non-profit entity and 70% to for-profit entities. There is a little definition of what is an innovation action. It's really to produce plans and arrangements or design for new, altered, or improved products, processes, and services. Remember the TRL 5 to 7. Now, evaluation principles. There are 4 principles under which we always make this evaluation. We use fair and equal treatment, of course, among all proposals. The evaluation is based on the work program and the defined set of evaluation criteria. Our experts are independent. They are selected according to their skills and knowledge. We try to ensure a certain gender balance so that there is no gender underrepresented. There is also a turnover every year, so we have to change these experts. They cannot work for us forever. There is a capping limit. And also to ensure a certain geographical diversity, and as well to include experts from the private sector, not necessarily from the public sector. And we also make sure everything is kept confidential, and we have a system in place to avoid conflict of interest. In the next slide, we can tackle the three evaluation or award criteria. You know them by heart, I'm pretty sure: the excellence, the impact, and the quality of efficiency and implementations. Each of these three evaluation criteria has a sub-criteria, and basically it's important to say that the evaluators will assess proposals against the topic text, against the 6.1 topic text, and using these evaluation criteria. No other sources will be used by the experts, by the evaluators, to assess the proposals. This is the definition of the scores. They go from 0 to 5, from a very super bad to excellent. Half scores can be given. If you see already 3 is good, it becomes very competitive. It's a very competitive call. These are the scoring thresholds. I'm sure you also know by heart. Pay attention to the impact because there is a correction factor of 1.5. So the scores, the ranking is made on a normalized scores, normalized total scores, taking into account that impact has 1.5. So if you want, it's not out of 15, it's out of 17.5. But in case of identical scores, precisely because this is an innovation action, the further ranking is done on the impact first and then on the excellence. So it's a competitive call. The available funding is a lot, in this case 74 million, but still we expect many proposals, and not all of these very valid proposals will attain the available funding. So very good proposals do not necessarily get funding. At the same time, what we say to everyone, don't waste your time if you're not 100% sure that you can deliver what the call wants. Understand call conditions and text. First of all, check the eligibility conditions. Then understand also the language that is used. At least, for example, doesn't mean all. For example, gives room for more. Also, make use of the national contact point of Horizon 2020. There are at least one of them in every EU and associated countries dealing with food and agriculture and bioeconomy. And also consult the FAQ system. Pay attention to the word, as I say. Shall and will means have to. Should, it means you will not get a good score if you don't do it. Then, when you are planning the proposals, leave no doubt on the TRL, technology readiness level, and the state of the art. Start in the situation now, what is the state of the art, and also what will it be at the end of the project. We need to substantiate this transition. Secondly, the impact is essential more and more because it is an innovation action. So you have to substantiate the impacts, how you're going to achieve this impact. It has to be credible. At the same time, we also want to see a dissemination and exploitation plan. Okay, we don't look for research and innovation action where a lot of research outputs are, let's say, produced. But at the same time, we want to see the exploitation plan, how the consortium is going to make use, commercial use even, of the achieved IPR. And also remember that 1.5 weighting for innovation action is key, precisely because it can make a big difference in the final ranking. On the next slide, we can see the implementation. Your proposal has to have a reasonable duration. As I said, the target is for 2030. If you plan a project that will last 15 years, well, I don't even think it's possible. I'm just making an absurd example. It's going to be a bit too late. You also need to justify your budget. Don't overestimate. And of course, take into account all the possible risk. For example, sources of external funding might disappear. The demonstration of this demonstration site, you will not get an access. COVID-20, COVID-21, COVID-22, who knows? And also the consortium Size does not matter, sorry for the joke, but it's true in this particular case. So big doesn't mean better. So just to cover the enough coverage that you need, nothing more. Then at the same time, you can have make use of graphics. Graphics help to explain, make sure, of course, they are readable. Evaluators have many proposals to read and not enough time, so your proposal text has to be easy to follow. And do not use tiny fonts or tight line spacing. Respect the page limits, and also write for non-native English speaker like myself. Why? Because we do not necessarily select all experts from that are English native speaker. On the next slide, I also want to touch about the cross-cutting issues. As said, a holistic approach is necessitated. So we pay a lot of attention to the fact that the proposal is interdisciplinary. So try to make a consortium that can cover all the various disciplines, also data, techniques, tools, and perspective. Take into account that there is also the stakeholders play an important role, their knowledge, and also the end users have to be involved. So not only academia, to make it short, or industry, but also farmers, patients group, consumer organizations. There is also a component of public engagement and social innovation. This is about to co-create the future with the citizens and civil society organization. And of course, do not reinvent the wheel, make use of existing EU initiatives, services, and projects. In this example, it's Copernicus and Galileo, but who knows, even results obtained by Mycokey projects. On the next slide, I want to just tackle quickly the international cooperation. As you know very well, Mycokey, but also my toolbox of Rudy, who presented before, had a focus on China. In general, the farm to fork topic has a focus on international cooperation. You do not need to look for a participant that is necessarily based in South America, Asia, or Africa. It's not an eligibility criteria in this case, but remember that this topic is flagged 6.1, it's in red below, as international cooperation. As a result, if you include applicants coming from specific third countries or international organizations, like some UN bodies, they are not necessarily funded. By default, our legal rules tell us that we do not fund them. But experts can consider that these applicants can be exceptionally funded. So if there is why, there is only one motivation for that: if the participation is deemed to be essential to carry out the action. Pay attention in the proposal template, because at the implementation part, there is a section on the consortium composition, and it is said, if you add an applicant that falls into this category, international organizations and applicant from industrialized countries or emerging economies, you need to explain why the participation it is deemed essential. Otherwise, the evaluators will very likely discard this participation, and perhaps the proposal is funded, but you will not have funding available for that particular applicant. A slide on open research and data management. So as you know, every peer-reviewed proposal that is published has to be prepared in open access. So there is online access to research results. You can decide to opt in or opt out in the open research data pilot. By default, the proposal tells you that you are opting in, but you can decide to opt out. The goal is, of course, to boost the benefits and maximize the impact of the research and innovation funding. There are things to take into account when you prepare the proposals. For example, you need to take into account that the cost for open access green or gold open access and data management are covered and eligible for reimbursement. And at the same time, you should also explain what's your strategy for open research data pilot and open access in the section 2.2 measure to maximize impacts of the part B. Then we always carry out an ethics screening, but this does not prevent you to say if your proposal is compliant with fundamental ethical principles, and you can also say what you want to do. There is an ethics self-assessment, and almost over. This is a slide with all the links, important links to existing projects, the Cordis, EASME Data Hub, INEA Data Hub, the Horizon Results Platform, Horizon Results Booster, and so on. This is more legal stuff. So H2020 Online Manual, there is a new section to find partners, and also a section on open access, data management, and international corporations, FAQ, and of course, national contact point and research inquiry service. If you have any questions, don't hesitate to write these questions to the research inquiry service or to address your queries to the national contact point. And of course, there is a huge list of FAQs that could have already the answer to your question. I am done. I don't know if there are any questions.
Thank you very much, Alessio. Right now, no question from the audience. Just Rudy ask us to just say thanks to you for your excellent guidance as a member of the commission during his project, and maybe he will do again personally when he will have again towards the start of the General Assembly. There is a comment, Alessandro, please.
Yeah, there is a comment from Logrieco. It is suggested the coordination, but by a private company.
Sorry, I didn't get it. The coordination.
The comment is the comment question is seems that there is a suggestion to coordinate to let coordinate the consortium for this project by a private company. Is that correct, or?
It's possible indeed. It's possible to anyone can be the coordinator. Just remember that as it is an innovation action, profit-making entities gets funded 70%. So usually the coordinator manages quite a lot of budget. So it should be a coordinator that is is regulated by private law, but it's not for profit. I think it would make more sense.
Okay.
It's not impossible.
Thank you very much, Alessio. And I think many questions will arise by maybe looking better the presentation you did, and surely you will have several questions by people of the audience. Now is my great pleasure to give the words and to invite Franz Verstraete from the Commission to have his presentation. The title of the presentation of Franz is EU Policy on Mycotoxins in Feed and Food, Update and Outlook. Please, Franz, you can share your screening we have your sharing already, your screen already shared. You need just to launch your presentation. And maybe you should click on microphone. No,
okay.
On microphone.
Yeah, the microphone would be okay.
Yeah, now it's perfect.
Yeah, you can hear me. Yes. Okay.
Yes.
So and you can also see my my presentation, I suppose. Yeah.
You need only the modality. Okay, perfect. Fine.
Yeah. Okay. So yeah, thank you for inviting me at this conference, and also to give you an update on EU policy on mycotoxins in feed and food. So important is that we are indeed regulating fusarium mycotoxins like deoxynivalenol, fusarium, fumonisin, zearalenone, T2 and AC2 toxin. And indeed, what we can see, and it's already regularly also pointed at several presentations during this conference, is indeed we see an increased prevalence following indeed climate change or also changing weather conditions. We can also see, and that is a major issue from a regulatory point of view, following this issue, this influence of the climate, the weather conditions, also a huge year-to-year variation. Some years you can find high levels of contamination. So indeed, then what we also can see, and fortunately also as well the Mycokey and as well also the My Toolbox has provided additional tools to the farmer for application in practice to prevent the presence of mycotoxins in food. However, we can see, and of course there are efforts done, but there is still further efforts needed to be done to have a good take-up by the farmers to use the mitigation tools available in practice. So there has to be incentives for the farmer to indeed effectively apply these mitigation tools, and also, of course, not only the farmers but also the storage keepers, the processors, and so on. Also, when we are discussing now, we have launched a new discussion round on regulation on mycotoxins in feed and food, and one of the issues is also that, as indeed you have already heard, we have also to take into account the possible consequences of policy initiatives which are under the EU Green Deal, and like for instance the Farm to Fork strategy. We see on the one hand, indeed, one of the targets is to have by 2030 a reduction of 50% use of risk of pesticides. We know that the use of, we have also seen some presentations that the use of certain plant protection products, fungicides, can also be helpful in mitigating the risk from the formation of mycotoxins, and indeed is one of the prevention tools. So this will also be a challenge. On the one hand, indeed, the need to reduce the use and the risk of indeed pest plant protection products. On the other hand, also we see this as we have also biodiversity strategy, and we know also, for instance, one of the risk factors is, for instance, the inoculum, which is present in non-cultivated areas alongside the fields, where indeed there is a requirement for biodiversity to keep indeed a non-cultivated zone close to, for instance, waterways and so on. But they are also, for instance, in ergot alkaloids, known to be indeed an inoculum for indeed infecting the field. So these are issues which are known and which we have also to take into account when regulating these fusarium toxins. So it's clear from indeed a lot of EFSA opinions and also a lot of other publications that there is a need to regulate fusarium mycotoxins in particular, but also other mycotoxins in feed and food to ensure a high level of human health and animal health protection. But of course, we have to also take into account the above mentioned considerations. So this is in fact as well applicable for feed and food. My presentation now will indeed first deal with food, and I will go through a number of contaminants or mycotoxins which are currently under discussion, and then in the second part I will also focus on feed and try to indeed not to be too long in order to not to already I would say aggravate the delay in the program. So as regards deoxynivalenol, so we have existing maximum levels in food already since 2006, but we are currently reviewing them based upon an EFSA opinion already back a few years ago, 2-3 years ago. And indeed, what we were looking is to set maximum levels not only on deoxynivalenol, but also on the sum of deoxynivalenol and the modified form, the 3-acetyl and the 15-acetyl deoxynivalenol, and also the DON-3-glucoside. However, when discussing, we have seen that although the analytical tools exist to analyze them, we can see that on a routine basis, they are not yet there widespread in, for instance, a lot of control laboratories. So there is still indeed work to be done by the EURL on indeed getting these methods applicable on a wide scale on a routine basis. And also we could see from the EFSA database that there are indeed a few, well, that are not sufficient data across all cereal and cereal products to really have an estimate of, okay, what is the sum of these deoxynivalenol. Indeed, in the EFSA opinion, as you can see, they have looked into it and they have done really made a simulation and have indeed calculated that they say on average, indeed, there is this 10% of 3-acetyl deoxynivalenol, 15% of acetyl deoxynivalenol, and 20% of DON-3-glucoside. But of course, there is a huge variation in concentration ratios. And when we also discussed that, also member states indeed indicated that indeed there are studies which indicate much different concentrations. And we also have seen in the presentation, for instance, from David Miller yesterday, that before yesterday on Monday, that indeed there is a huge variation, can be from a region, can be year-to-year variation, that there is indeed a huge variation as regards the ratio of DON-3-glucoside or 3-acetyl deoxynivalenol or 15-deoxynivalenol, acetyl deoxynivalenol versus the parent compound. Yeah. So therefore, but okay, we have seen from an enforcement point of view, we must have indeed clear possibilities for routine methods of analysis, providing reliable results. And we have also to base our levels, we have to know exactly also sufficient data on the sum of the deoxynivalenol. So these two factors are not yet fully up to date, and therefore we have for the time being decided to review the maximum levels based currently also again on the parent compound. But on the other hand, we have really we really recognize the need to really regulate the sum of them, because they are from a toxic point of view as toxic as the parent compound. And so we will also launch an exercise to gather in the coming years sufficient data for possible regulation in the future. And now we are currently reviewing the maximum levels based upon what is as low as reasonable achievable based upon recent current occurrence data. We can see that we can effectively lower the current maximum levels, but of course we have also to take into account the issues which I said at the beginning of my presentation, and that's clearly still indeed in discussion with stakeholders, with indeed representatives of farmer organizations and of processors. So indeed, where we are with this review of these maximum levels, we have already done a targeted stakeholder consultation. We have had also already not what they call a forum, but indeed a thematic session of member states and stakeholders to discuss the issue in detail. And so we will now already from next Monday start the discussions to look into the comments received and to try to finalize the technical discussions on the review of the maximum levels for deoxynivalenol by the end of this year or beginning of next year. Also in T2-HT2 toxin, for the time being, we have only indicative levels in a Commission recommendation, 2013. But since then, we have seen that EFSA has reviewed the health-based guidance value, and also the exposure has increased, also due to the indeed increased prevalence in our food. And so therefore, we are indeed now considering to set maximum levels for the sum of T2 and HT2 toxins in cereals and cereal products. And of course, as I said, we also have to look into the issues relevant for deoxynivalenol, the year-to-year variation, the issues related to geographical issues and so on. But it can be said that what we are currently discussing as regards maximum levels, they are lower than the indicative levels as they were in 2013, 165. So also there we are moving to a stricter approach as regards the presence of T2 and H2 toxin in food. Also there we are exactly at the same timing as regards deoxynivalenol. We had also targeted stakeholder consultation, and we are currently now, from next week onwards, discussing to finalize from a technical point of view the proposals of the maximum levels for T2 and H2 toxin. Then moving back to what is already decided, is citrinine is in fact a particular issue. We have currently only it regulated in rest yeast rice supplements, but just to inform you that we have lowered the maximum level from indeed what was existing from 2000 microgram per kilogram already in 2014. We have now lowered it last year to 100 microgram per kilogram, so a factor of 20 lower. Indeed, and this level applies since April of this year. Another hot topic is indeed the ergot alkaloids and ergot sclerosia, which is also mycotoxins from indeed claviceps, but indeed not as well, not in the focus of my toolbox, neither of Mycokey. So we have currently a maximum level for ergot sclerosia, that are the sclerosia present in unground cereals, the purple parts. And there we had a current level of 0.5 gram per kilogram for food. We are considering to lower it to 0.2. But then more importantly, we are also discussing the setting of maximum levels for ergot alkaloids, the sum of 12 ergot alkaloids and the enes and the enine forms. And the maximum levels are lower bound. So therefore, we also establish specific limit of quantifications to be respected for the normal milling products. Limit of quantification of is 4 microgram per kilogram. But for cereal-based foods for infants and young children, the limit of quantification is 2 microgram per kilogram. Also, and that is also an important issue which we discussed and which we for the time being have no provisions, the similar provisions in other mycotoxins, is that for the first time we have set separate levels for indeed what we would call whole meal flours and white flours, or indeed, for instance, also high fiber breakfast cereals versus low fiber breakfast cereals. The point was that there was no criterion to really distinguish what is a whole meal flour and what is a white flour. Okay, the extremes, they are clear, but you have a lot of intermediate products which would not be made clear. But indeed the stakeholder, the European flour millers, they have come forward with indeed a criterion, the ash content, which is indeed could make the separation, and therefore we will indeed set a higher level for indeed the what we call the whole meal flours, because indeed, as you know, the mycotoxins mainly occur on the bran, on the outer side of the cereals, than indeed for the white flours. So also ochratoxin A, we have in fact already extensive legislation in the EU since 2002, but we have a recent EFSA opinion from 31st March 2020. And of course, one of the issues is still the discussion on the genotoxicity. Of course, it's clearly ochratoxin A is a carcinogen, but the genotoxic nature is a bit still under discussion. However, EFSA, based on the weight of evidence, is of the opinion that it could be considered as a genotoxic carcinogen, and has therefore applied the margin of exposure approach. And they could see that with the current exposure, with the current regulated levels, that we still have an exposure which is indeed results in margin of exposures lower than 10,000, and consequently are indeed of a possible health concern. For that reason, we are indeed currently discussing to set maximum levels for indeed a number of food commodities which are not yet covered, and also to review a number of maximum levels. So as regards what are the maximum levels we are currently considering to set in addition to the existing ones is for dried figs, also for dried white fruit other than dried figs and dried vine fruit, because we have already a level for dried vine fruit. We will now cover all spices, also licorice for direct human consumption. A bit strange also, we could see quite some significant levels in dried herbs and tea and herbal infusions. So a lot of them do not contain any ochratoxin A, but then certain teas do contain significant levels. We will also set levels in a number of indeed oil seeds, like indeed sunflower seeds, pumpkin seeds, melon seeds, hemp seeds, and also soybeans, and also from the tree nuts. We were discussing to set the eventual, and finally we will only set levels for pistachios. At a certain moment, hazelnuts was also in the discussion, but finally was found not necessary to set a maximum level for hazelnuts, and also for cacao powder for human consumption. Besides the setting of these new levels, because they're commodities which are not yet covered by EU legislation, we are also considering the review of existing levels, and that's particularly for dried fine fruit, where we have currently a level of 10 microgram per kilogram, and the level under consideration now is 8 microgram per kilogram, and would be the same for dried figs. For roasted coffee beans and instant coffee, we could see that in certain countries, this is the main significant contributor to the exposure. We could see that from biomonitoring data, that indeed the highest levels in the biological matrix was found in people consuming a lot of coffee. So we will indeed also lower it's foreseen to lower the levels for roasted coffees from currently indeed the level of 5 microgram per kilogram to 3 microgram per kilogram, and for indeed instant coffee from 10 microgram per kilogram to 5 microgram per kilogram. And also for the cereal products, which also including the fine bakery wares and bread and so on, we will also set a stricter level than the current level of 3 microgram per kilogram. Also, what we have seen, but of course there are not so many data in the EFSA database, that there was also an issue with potential presence of ochratoxin A in cheese and ham, but there it's also an issue of indeed sampling, because you can in a ham as such you can find indeed higher levels at the outer side and lower levels in the core of the ham. So there has to be an issue as regards sampling also. As regards the timing, there we will launch the targeted stakeholder consultation on the suggested levels in the coming days, and then we will see what comments we receive, and we will continue the discussions on technical basis with member states early next year, in view of finalization of it in the course of next year. Oh, we have to moment. Yeah. So then, as regards aflatoxins, so there also EFSA has a recent opinion, and also there we have an extensive EU legislation, and also one of the most strictest legislations in the world as regards aflatoxins in several foodstuffs. But indeed, as you can see in the EFSA opinion, even with the strict legislation, EFSA came to the conclusion that the current exposure to aflatoxins in the EU population still raises a health concern. We still see an issue of indeed that the cancer risks, and as you know, indeed the Commission is also working on a plan beating a cancer plan, beating the cancer, and also indeed a reinforced approach as regards carcinogenic contaminants is therein foreseen. So indeed we will have to see as regards, even if we have a strict legislation, what indeed we should do as regards to reduce the further risks as regards the presence of aflatoxin in food. We have started the discussion with the member states. Following this opinion, we can cover indeed already a bit of number of foods where we can see that there are some significant levels of aflatoxins in herbs, a bit the same as for ochratoxin A, but also covering all spices. Also, we could see that also in cocoa that there could be an issue of aflatoxins. And there was also a request, although we have already existing maximum levels for almonds, which are in line with Codex, but there was also a request to effectively reduce the maximum level, as indeed some exposure estimates have indicated that the exposure from aflatoxins due to the consumption of almonds was considered to be too high. As regards the timing, also there we are still discussing it. So I think it will be only in the beginning of next year that we will be able to consult stakeholders on our suggested levels, and we can then finalize the technical discussions and the regulatory procedure in the second half of next year. So with finalization by the end of next year. On alternaria toxins, well, in fact, as you know, the issue is that EFSA got alternariol and alternariol monomethyl ether. They also identified that this could be indeed the genotoxic carcinogens, and indeed for and that, but there are no toxicity data available to do a comprehensive risk assessment. And this was already identified in 2011. And in fact, still now, 9 years later, there are still not any relevant studies which would enable to do a comprehensive risk assessment and to really have a good idea on the risk. The only approach is that they could follow according to the tool of the threshold of toxicological concern. They could say, well, the presence of indeed alternariol and alternariol monomethyl ether could be of a potential concern because indeed the threshold of toxicological concern is exceeded, and also for tinoazonic acid, which is not a genotoxic carcinogen, but indeed also there the threshold of toxicological concern was exceeded and could be of a potential health concern. But they could not conclude on the real health concern because there are no toxicity data since already now 9 years. On the other hand, they could clearly, based upon this threshold of toxicological concern, say that tentoxin, which is the other alternaria toxin, that is not of concern. So also we are confronted, of course, we have no comprehensive risk assessment, but indeed there is pressure from the member states to also regulate alternaria toxins. We need indeed more sensitive methods of analysis and also more data on the occurrence, and also on seeing what indeed what are the good practices to avoid the presence of alternaria toxins, because there is also not so much information available on this. And so there we are currently finalizing a recommendation to monitor the presence, to also identify the factors which are resulting in the presence of alternaria toxins, and indeed also in order to trigger these investigations, we have set also indicative levels from which we think the investigations on the sources should be performed. And these indicative levels, they have been set for alternariol monomethyl ether, and also for indeed tinoazonic acid in indeed processed tomato products, paprika powder, sesame seeds, sunflower oil, tree nuts, dried figs, and also cereal-based foods for infants and young children. So there we have also put in specific analytical requirements, because what we could see that with the current methodologies used in routine control, that indeed the sensitivity was not enough. So there was a lot of indeed left censored data demonstrating that the sensitivity was not enough. Now moving forward quickly to feed, and which I will expect the current ongoing discussions, but also two aspects as regards the criteria for detoxification processes, and also the requirements for mycotoxin binders and detoxifying agents, which is also of relevance in the frame of this research project. So as regards mycotoxins in feed, we have a bit of a complex regulatory situation. We have maximum levels, which are comparable with the maximum levels in food in the Directive 2002/32 EC. We have a more flexible or indeed less strict regulatory approach as regards all the other mycotoxins, so ochratoxin A, zearalenone, deoxynivalenol, T2N, C2 toxin, and fumonisins in feed by setting guidance levels in a Commission recommendation. So this is a soft regulatory approach. And then also we had still these indicative values for T2N, C2 toxin. However, we are currently discussing a complete new approach for regulating mycotoxins also in feed, whereby we would say, well, indeed, we have for a number of these mycotoxins reference points for adverse animal health effects, which have been identified by EFSA and also in other studies. And indeed, these levels would be the maximum levels in the levels in complete feed would reflect these reference points for animal health and adverse animal health effects, which would mean that if indeed the levels in the complete feed are exceeded, that they could result in adverse animal health effects, and therefore it was considered to be appropriate to indeed to have also the levels in complete feeds to be having set it as a maximum level. So a much more stricter regulatory approach. While for feed materials, there we would still follow the more, I would say, softer approach of guidance levels. Indeed, also to take into account the huge year-on-year variation, and indeed, and also because there is a management, there is a different sensitivity of animals to mycotoxins. So you could then, the higher contaminated, for instance, the higher contaminated feed material of deoxynivalenol, you could use it for instance, for bovine, for ruminants, which are much more tolerant than, for instance, the pigs. For complementary feed, we would follow an approach of an action levels, whereby an action should be undertaken when they are exceeded. So this is a new approach which we are currently discussing and working out. So as regards then deoxynivalenol, indeed we would set anyway lower levels for guidance levels for feed materials. We would set also, we have seen in the recent years that there has been an issue with deoxynivalenol also in sugar beet products for feed, and also whatever levels it will be, maximum levels or guidance levels, we will also have to lower, based upon the recent EFSA opinion, the current guidance levels for complementary feed and compound and complete feed. For T2 and HT2 toxin, while we have currently only indicative levels in the frame of a Commission recommendation, also there we are discussing the setting of maximum levels for complete feeds and guidance levels for feed materials based on the sum of T2 and HT2 toxin. For zearalenone, also there we the same approach, possible maximum levels for complete feeds, but we also looking at setting an additional level for sugar beet products, but also for oilseeds and derived products, because we could see that also zearalenone could be present at significant levels as well in sugar beet products, as well in oilseed products. And also we are looking into lowering the guidance levels, current guidance levels for cereals and cereal products, and including maize and maize byproducts. And for fumonisin, also there we are currently reviewing the current guidance levels for maize and maize products, and also significantly lowering them, in fact, because we can see that from the occurrence data that they are way too high. And as regards the levels for compound feeds, there are currently slight modifications under discussion. When I speak of slight modifications, it's always indeed lowering the levels. We are not planning to increase any of the levels. So also there in the feed we also discussed the ergot issue, and there we had also a maximum level of 1 gram per kilogram unground cereals. There is discussion to lower it to 0.5, and also we are currently discussing to set regulatory levels for the sum of ergot alkaloids also in feed, possibly for to begin off with indeed guidance levels, as we have indeed first to see if it is a real issue as regards the presence of ergot alkaloids. Then I would like to just highlight the issue of regards to feed criteria for detoxification processes, because there have been indeed as well in both research projects been processes identified to detoxify feeds, and it's important that indeed for feed it is possible to detoxify. It was already foreseen in Article 8 of the Directive 2002/32 EC, with the possibility to set acceptability criteria. In the Commission Regulation 2015, we have set these criteria for the detoxification processes, to which indeed the acceptability criteria for such a detoxification process. It is also foreseen that indeed, well, it is indeed some additional provisions, but important is that such a detoxification process can only be applied if EFSA has performed, on the request of the Commission, a scientific assessment of the detoxification process, concluding that the detoxification process complies with the acceptability criteria set out in Commission Regulation 2015/786. And this applies already since the 1st of July 2017. For the time being, there has been in the past an application for the detoxification of aflatoxin by ammonia, but this has not been pursued because indeed the applicant was required to provide additional information which he was not able to provide, and finally the assessment was stopped. There is now a new application also for indeed the decontamination of indeed groundnut meal by ammonia for aflatoxins. But for the time being, there's no detoxification process for the presence of mycotoxins in feed assessed by EFSA with favorable outcome, which means that in practice in the EU, no detoxification process can be applied with as regards contaminated feed materials, as regards mycotoxins. There are a number of detoxification processes favorably assessed for dioxins and dioxin-like PCBs, also for hydrocyanic acid, but not for mycotoxins. Another issue is also a point is the issue of the mycotoxin binders, the detoxifying agents, for which indeed back in 2009 we have established a new functional group of feed additives in the category of technological additives. It means also that indeed before being able to be used in feed, that these substances have to be authorized at EU level prior to use in feed. It's also important that indeed it's not only important to develop these techniques in research projects and to work out, and indeed they are showing favorable results, but also the next step, someone has to take it on zich and indeed make an application for indeed having it authorized at EU level for use in feed in the EU. And so indeed important is what is the difference between these binders and detoxifying agents compared to indeed the detoxification process. Well, in fact, these binders and detoxifying agents have to be used in feed compliant with EU legislation. So it's important that they comply with the feed in which they are applied, that they comply with EU legislation. It is also not the aim for the time being to indeed set maximum levels higher than what is indeed would be allowed from an animal health point of view, saying okay, you can apply these binders which mitigate the animal health risks. This is not the case for the time being. So but of course the use of it is very important because it can provide additional guarantees, and indeed these additional guarantees, there is, well, we know that mycotoxins are very heterogeneously contaminated in a lot, so it's indeed to mitigate the risks related to a heterogeneous contamination. There's also when you have a year with a high prevalence, indeed by sampling you cannot detect all the non-compliant consignments, also because of this heterogeneity. Also, another issue is that for the time being we have set only levels for individual mycotoxins. We have not yet indeed considered the combined exposure, while we know that, for instance, a lot of cereals and maize can contain simultaneously high levels of deoxynivalenol, high levels of zearalenone, and high levels of fumonisin. And so indeed it is also the use of these binders can be very helpful indeed to mitigate the risks as regards to combined exposure. Also, as I said, we have not in legislation yet fully taken into account the modified mycotoxins. Also, this is indeed could be indeed appropriate to use it, and we know that indeed we set these levels and for indeed an animal category, but within an animal category there is a difference in sensitivity. There are sometimes very sensitive breeds, for instance of pigs, to deoxynivalenol. And so also for these ones, it could be useful to add indeed the binders or the detoxifying agents to indeed ensure that there are no animal health adverse animal health effects occur. And so what are the currently authorized mycotoxin binders or detoxifying agents? So we have one absorbing agent, the bentonite, the aflatoxin B1, and we have indeed currently authorized 3 mycotoxin biotransforming agents, which is indeed a microorganism and a bacterial strain from the Corynebacteriaceae family for trichotillomers, as well in pigs and all avian species. And we have also 2 different fumonisin esterases, one produced by indeed Comagatella pastoris and the other one by Paffii, and both in fact for pigs and avian species. But these are also the only authorized ones. So if there are new indeed biotransforming agents or absorbing agents developed before they can be used at in the EU, they have to go through the authorization process after an assessment by EFSA. Important to highlight this. So to conclude, I make it very short, is indeed that regulating mycotoxins in feed and food is important and necessary for ensuring a high level of animal and public health protection, but it's challenging. And I hope indeed that the mitigation tools and the prevention measures which have been developed and identified and worked out within the Mycokey research project and also my tool research project will help us to indeed ensure this high level of animal and public health protection. Thank you very much for your attention.
Thank you very much, Franz, for this really global overview of the current regulations, the current issues on the mycotoxin in food and feed. I think we had a very excellent conclusion of this 3 days with your talk, so I really appreciate, like all audience surely did, and I'm sure that soon there will be further chance to interact about these issues. Now I must, because we are really running out of the time, give the words to Antonio Greco for the final conclusion of this scientific 3 days. Thanking again our last but not least speaker, and thanking again all the sponsors that support us for this conference online and that will support us for our physical conference. But just a question to Franz. Franz, please.
Okay, a question from Prodi Antonio, which is the idea regarding the legislative regulation of emerging mycotoxins such as bevaricins and niacins?
Well, yeah, that is also a point that is on our radar. We have not yet any concrete discussions. We had an issue with niacins to really start to regulate it, but for the time being we have not pursued this, also because of other priorities. But it's certainly the niacins is certainly an issue which we have to look closer in, because we can see that sometimes very high levels have been found, certainly in certain regions of the EU. So yes, it is also on the radar, but there are no concrete discussions ongoing for the time being. But yeah, even if I would put an outlook, they would be put on the outlook. Yes.
Okay, thank you very much again, and I say again thanks to all speakers, and then I give the words to Antonio.
Thank you very much, Antonio, and a special thanks to Franz for his excellent talk. So this is, we have at the end of the 3 days of online Mycokey conference, and I'm quite satisfied about how this conference was running. So of course, I like to thank a lot of people about how technically was conducted this conference. I have to thank Mimmo, Cenchi, Alessandra, Villani, and Mariela Quarto. But also I'd like to thank, as I spoke at the beginning of my talk, a lot of people that play important role in conducting this important project, European project. So all speakers, of course, but also all players, all researchers that actively interact with each other. Of course, all work package leaders that really conducted and coordinated well own work package. And then also is a particular thanks to External Advisory Board, conceived by David Miller from California University of Canada, Christopher von Holst from European Commission DGRC, Serena Borgna from Agency of Promotion of European Research Italy, Franz Lestrade from DG Sanco, and Peter Karlowski from George August University from Germany. So they really help us to improve, and to adjust some activity in order to reach the specific goal that we wrote in the proposal. So Franz, he talked specifically about the importance of regulation at European level. And I'd like to take this opportunity, this chance to introduce just a small presentation about one recent project approved by European Commission dealing about of this direction, about the importance of the regulation at the European level, particularly for contaminant, including mycotoxin, and then how this project can be linked between the European Commission, European EFSA, and all stakeholders, officer, the different players along the chain. So I'd like to give a talk, the awards to Veronica Lattanzio, that she is coordinating this project, just to updating about the action and the activity about this proposal just approved. Please, Veronica.
Okay, mi vedi? Okay. Hello, can you see me? Can you hear me?
Yes, you go ahead.
Okay, so I just share my screen. And so I thank you, Dr. Logrieco, to give me just few seconds to introduce you our new adventure, the new adventure that Ispa is embarking soon after the Mycokey project. Is the Food Safety for EU project, a multi-stakeholder platform for food safety in Europe. This is a project that has been recently founded by the European Commission and under the Horizon 2020 program and under the topic supporting the food safety system for the future. Is a coordination and support action with a budget of 3 million euros, and we stay for 3 years starting from January 2020. We just signed the grant agreement. So the Food Safety for EU project will design, develop and release a multi-stakeholder platform for the future European food safety system. This platform is intended as a participatory place, which will be virtual and physical, and will employ new digital tool. The platform aiming at sustaining the food safety actor through a multi-actor approach, so sustaining all the food safety system actors to become a community which will be inclusive, adaptive and responsive, even to fast changes scenarios. This community will support and hopefully will work in synergy with the food safety authorities, EU services, EU agencies, and policymakers. So the goal is to work through co-creation through co-creation to produce, to deliver a new framework for future policies by alignment or research program, either at national and transnational level, by proposing a new strategic research and innovation agenda, by drafting new models to boost the public confidence in the European food safety system. The project, the platform, and this community will support EFSA in achieving, in improving transparent communication, and also by ensuring access to data, information, and knowledge. The consortium at the moment is pooled by 23 partners from European and associated countries, but also involve a wide network of about 80 external experts that will be involved in the participatory process, 6 independent advisors, which will support us in aligning our strategy to the European policies and the main goal, and also involves 44 supporting partners at the moment, which signed already their commitment as a stakeholder, and include 12 food safety authorities, 22 research centers and producers association at country level, and 10 among food industry association and consumer association, also some other EU organization and federation. And also we are on the way on establishing liaisons with other European projects which are ongoing to share background and to share objectives in order to avoid overlapping. The plan, the ambition is to enlarge this stakeholder network. So there is the possibility to cooperate with the project. So if you want to learn more, you can go through the Cordis website where there is a fact sheet and other information about the project, or you can contact directly us, myself or Dr. Logrieco, to see how you can contribute to the project. Thank you for your attention.
Thank you very much, Veronica, for your presentation. This is a big challenge that all scientific community, but together also with the different European agency, can have in the future, next future. Anyway, so I'd like just to make some final conclusion about over these 3 days, and then I'd like also to thanks, particular thanks to also to Alessio Bacchielli, that he really was very active, fruitful in his suggestion how we could conduct correctly all bureaucratic part of the project. He was very proactive. Every time he was very useful in terms of to find the right solution for each problem. And then, so of course, I have to express my sincere gratitude for Alessio Bacchielli. And also, I'd like just to thanks to really with the Rudy Cresca, that we had a really great cooperation with 2 different projects. At the beginning, we were, of course, competitive in terms of consortium, in terms of project, but once the 2 projects were funded, then we moved on the other sides of the river, and then we collaborate very, very, very active. And so in these 3 days, we showed our productivity in terms of outcomes, in terms of collaboration. Anyway, so this is, as Alessio point out before, so this is a European Commission has launched the European Green Deal to make the EU economically sustainable. And this is a different challenge, and that this is one of the major pillars is from farm to fork. This willingness to cover the importance of this aspect, to cover the whole chain, because particularly in the Green Deal, this is from farm to fork. This is the real magic possible solution. And then going in details, so we can find these key words is precision agriculture, organic farming, including the need to increase the area under organic farming, agroecology, stricter animal welfare standards. So this is the key sentence, the key words that from farm to fork, the European Green Deal include. So, and these, I'd like just to show you how Mycokey predict in this we can say predict this aspect in during the project, and we can anticipate part of this challenge. This is important key challenge in during the project. Particularly has contributed the Mycokey to use alternative plant protection products to reduce mycotoxin incidence in the crop and the impact of the human and animal health. And then we saw the nice introduction, nice presentation by Susan Wokelskan about all different approach in order to prevent the mycotoxin contamination in the field. So by using biocontrol, reducing the fusarium inoculum, by breeding for resistance, and so on. This is a really alternative plant protection products. This is just also as by reducing biopesticides. So Alessio emphasized the call about reducing pesticides, optimize fertilization. So this is one part is reducing pesticides by using biopesticides, of course. And then of course the big challenge that we carry out in Africa, as well as also in other parts in China and in Europe, has by using of very effective toxigenic formulation in the field. So this is really big challenge with really enormous impact in society and the economical aspect in this great continent as Africa is. Then also Mycokey has contributed significantly in precision agriculture, developing predictive models and making a more robust decision support system for management by farmers. This is another very important word, farmers, because the farmers play in the Green Deal is important role of mycotoxin mitigation with lower environmental impact. So we develop a different modeling, and then so also we use the importance. We studied the importance of biodiversity, about of toxigenic fungi at the global level, because by climate change, this topic will be one of the most important challenges in the future. How the climate change may influence, may interact with the toxigenic fungi distribution and of course the mycotoxin risk at the global level. So this is another also the density solution by Theo, by Mycokey app. This is an important tool together with also the My Toolbox tools to support the farmers in order to mitigate, in order to manage the problem in the field level. Then Mycokey has contributed the use of preventive tools such as development of a less susceptible resistant maize and the wheat variety to reduce mycotoxin fungi ability to colonize plants. This is the more attractive and more productive and more robust way to reduce, to minimize mycotoxin in the field. But of course, this action needs a lot of times, a lot of efforts, because to find resistance or less susceptible variety is a long time requirement. And then, but also by using the new technology, we can facilitate to reaching this goal. So this is just an example how we can find the right QTL for resistance in maize, and then also for durum wheat in China. So this is where very successful outcome of the project. So then we also developed several tools, additives and toxifying agents, to reducing the impact of mycotoxin on animal health, match the aim of farm-to-fork strategy of stricter animal welfare standard. This is another point, important point of European Green Deal. So in this aspect, in this context, we developed, we studied, we conducted different research towards to find out to solve, to obtain a new bioorganoclay solution in order to decontaminate for the contamination of mycotoxin in vitro, in vivo, with very active interaction with the industry. So this is another important part of the Mycokey. We had, as I said to you before at my talk, we had very high TRL successful outcomes about the project with different exploitation activities. So this is was due to the strict collaboration with the industry. And then also the remediation with also different approach with the using of enzyme, different microorganisms, and then is very promising, effective, and very applicable methods. So then the one more challenge that Mycokey, but both consortium carry out was the contribution in developing a wide, strong networking among scientists, institution, company, and stakeholder through several meeting, activity, appointment, demonstration, roundtable, etc. This for sharing the knowledge, because one of the work package in Mycokey was the knowledge, the global knowledge, because we have not reinvented the wheel, but we have to go on. We have to go forwards, new knowledge, new technology, and then we have to make the point, the update about all this knowledge and to go forward. And then this is important to organize, to facilitate this network in order to avoid overlapping in different projects, to avoid overlapping in different activity, regional, national, and international activity, because our goal is to solve the problem, to minimize the problem at the global level by considering a different, of course, location that is quite different geographical location, of course. So this is just some slides about how we can teach, how we can interact online as well as by physical approach, when it was possible. Okay, of course. So finally, take-home message. So this is these are not the usual sentence, but really we believe in this sentence. We believe in this message. The importance of the multidisciplinary action for combating the mycotoxin exposure. So this is a specific problem where all different disciplines have to interact strictly each other in order to know better the problems, the interaction, and how we can solve the problem. This is very important strategic part. Then is the importance of multi-approach along food feed chain from farm to fork, because as you can see during the project, it's important to be active along the chain on different steps, because as you know, the farms is important, the field is important, but also how we can reduce, how can minimize the mycotoxin impact also after post-harvest is a strategic and a crucial part of the game. Then the importance of the global cooperation for sharing knowledge, technology, and solution for a global problem. This is important. And then we were very happy about to sharing the knowledge with the Chinese consortium, because the China represents a very high competitive, but as well as but a contemporary very high productive in terms of knowledge and technology. And then it is important to sharing and to cooperate in these fields. But this was underlined, it was included in the code. So this is, we accepted. And then although the consortium were very consistent in terms of number, because we managed 11 partnership from Chinese consortium and 23 from a EU consortium. So based on a good organization, we really cooperate actively, and then so we conducted the games in excellent way. So interaction of mycotoxin, toxin fungi, crops or commodities, and climate change, I believe that this will be one of the most important priority in the future challenge. So this is the climate change is very, very important, will be or is a very important action parameter in order to spread and to adapt different population in different situation and with different challenge every year. And then so mycotoxin has one, it could be considered as one of the most crucial challenge for EU sustainable development goals. So these which include no poverty, zero hunger, good health and well-being, industry, innovation, and infrastructure. So this is I believe that mycotoxin is a very, very important and crucial challenge in the future. So we must, we need, we have to consider that we have to consider one health, because we are in only one world. So I think that this is mycotoxin problem really is a global concern, and then we need to having one health in terms of human, animal, plant, soil, ecological, so different aspects, that different key action, different key words that have to play all together in the same game in one word. Thank you very much for all your attention and for all your contribution and for all your audience.
Thank you very much, Antonio. Now I will say just with a few data, numbers about the conference, goodbye to everybody, and of course invite all people online to the ISM General Assembly. So you can see on my shoulder the images of our first kickoff, the images of the meetings that when it was possible that characterize also this 4 years of project, and how this project has mean also a lot of interaction, human interaction, and apart the scientific, very good scientific work together. So as an end of the 3 days project, I like to tell you then that we had 40 speakers, 6 scientific sections. We had over 3 industry sessions, more than 1,200 slides were shown, more than 1,000 participants in 3 days, 65 participant questions and answers, 180 people were online on YouTube, and the visualization on YouTube were 1,300. I think these numbers mean that all our conference was very highly appreciated. I thank you very much, everybody. And now I thank you, Antonio, for the strong coordination of this of this great project. He has led with great capability this project, and we achieved really a lot of work. And I would like to thank again Mycokey, Alessandra Villani, and all team of Ispa, Mariella Quarto, and all team of Ispa, Nunzia Cito mostly, which is our manager, that could not attend to these 3 days for a personal problem, but that has followed us and has been, I would say, the pillar of all this project. Thank you again. Thank you, Antonio. Thank you all. And I invite you to join us at the ISM General Assembly.